I'm Tom Risch, Vice Provost for Research and Technology Transfer here at Arkansas State University. Welcome to Create It State, our celebration of research, creativity, and scholarship by our students. Today, you're going to be watching presentations that have been done virtually due to the COVID-19 pandemic. We welcome you and hope you enjoy our students' research. And as you go through each presentation, keep in mind the hours of work that goes into these, not only by our students, but also by our faculty mentors. Today, you'll be seeing research presented by our students with both undergraduate and graduate. Also, as part of our endeavor, we have invited some high school students to take place that have been mentored by our graduate students. Research is part of our rich academic culture here at Arkansas State University, and it's these high impact practices where students work one-on-one with faculty members that we really feel is our signature of our academic excellence here at Arkansas State University. I hope you enjoy the presentations and look for another virtual presentation coming this fall. And then by next spring, we hope to have everybody on campus where we can have a celebration of 3 days of our students' research, creativity, and scholarship. Wolves up.
Hello, and welcome to Create It State 2020. This is the College of Sciences and Mathematics version of Create It State, and I am Lynn Boyd. I am the Dean of that college. I'm really excited to be involved in Create It State because this is going to be my first year being able to see it in person. You know, when I knew I was going to come to Arkansas State University, I heard a lot about Create It State and how great it was, and I'm really excited to actually be able to participate in it. Our college has a lot of really cool and exciting research going on with our faculty and our students. I'm excited that our students are getting engaged in that. You know, it's been documented that students who participate in research have better outcomes in college, and they are more likely to go on to graduate or professional schools. So it's a great opportunity that students have to do this, and I'm glad that they're taking advantage of it. In addition to the benefit to the student and the great student experience, research is really important for our society because through research, that's how we make advancements. That's how we discover new things and learn about how the world works. So, and I'm always telling students that those things that they read about in their textbooks, that wasn't put there by magic, but that it's people doing research who discover these things who then end up being our body of knowledge. And as a laboratory scientist myself for most of my career, I know that making new discoveries has really been the most exciting and rewarding part of my career. And especially sharing that with students and seeing students' eyes light up when they realize that all their hard work, all their time in the lab, and all their experiments finally has led them to make a discovery that nobody knew before. And that's really exciting, and I'm really looking forward to hearing from our students what kind of new discoveries they have made in their research. So with that, I think it's time to move on to the research presentation. Thank you so much for joining us.
My name is Clay Hearn. I'm a Bachelor of Science of Education and Mathematics student here at Arkansas State University, and I have recently conducted a study on the perceptions of Northeast Arkansas secondary mathematics teachers on the ability for students with learning disabilities to succeed in mathematics. And my mentor is Dr. Lisa Rice. I have personal connections with learning disabilities because I have nieces and nephews that have been diagnosed with dyscalculia and dyslexia, which are learning disabilities that affect learning mathematics and reading, respectively. And research suggests that teachers tend to have lower expectations of students who have learning disabilities in the field of mathematics. and this perception could potentially hinder the success of the student's mathematics education. There is also limited research on teaching mathematics to students with learning disabilities versus teaching them in reading and English. Lowering expectations of students with learning disabilities is often a result of viewing them through the deficit model, which is viewing individuals as their weaknesses rather than their strengths and environment in which they are a part of. And there is also a lack of research that deals with learning disabilities in the field of mathematics, which is going to result in a lack of intervention strategies backed by research that are readily available for math teachers. And however, despite this lack of research, studies show that students with moderate to severe intellectual disabilities are still able to learn mathematics beyond procedural fluency. This means that they're able to achieve a conceptual understanding of the material rather than only knowing how to go through the motions and get an answer. And it should also be noted that special education teachers often don't have the education or training to prepare them to teach math. Along the same lines, math teachers don't have the education or training to prepare them for teaching special education. So this led me to develop two questions that guided this study, and these are: what are math teachers' perceptions of students with learning disabilities? And what are teachers' expectations regarding success in mathematics for students with learning disabilities? So the study serves the purpose of gathering themes regarding learning disabilities in terms of general and math-exclusive learning disabilities and other factors that may hinder learning. It also serves the purpose of gathering themes regarding teachers' expectations of students' ability to succeed in mathematical problem solving. To answer these questions, a survey was developed and sent out to secondary mathematics teachers in this region, and it contains 3 items. Demographic questions were to gather data on the background of the teachers. Likert scale questions were given to give an indication of how confident the participants feel with teaching students with learning disabilities and their expectations of students with learning disabilities. And open-ended questions, which allows the participant to further explain strategies that they have found to be most effective in teaching students with learning disabilities. This survey revealed that these teachers do have high expectations of students with learning disabilities. However, they don't feel like they've been adequately prepared to teach these students. And it also showed that breaking the lesson into smaller chunks to make them more manageable and less overwhelming is a common strategy that helps these students with learning disabilities. And many schools haven't adopted intervention strategies or a program to help students with learning disabilities to learn mathematics. Based upon the survey responses, these teachers have high expectations of students with learning disabilities, which conflicts with the deficit model, which was a common theme throughout the literature. However, in contrast, in support of the literature, these teachers feel unprepared to teach mathematics to students with learning disabilities. So this shows that professional development and strategies that are backed by research regarding teaching students with learning disabilities need to be provided to math teachers. This would be done so by further researching on learning disabilities exclusive to mathematics. The importance of this study is to raise awareness of gaps in understanding secondary education learning disabilities specific to mathematics, and the need to develop mechanisms to address such inequities in the learning environment, ensuring that students are given the resources that they need to succeed in the classroom. Before concluding, I would like to thank you all for attending my presentation, my thesis committee members, Dr. Lisa Rice, Dr. Amanda Lambertis, and Dr. Farabe Tano for their guidance, Northeast Arkansas secondary mathematics teachers for their willingness to participate in the study, and my family and friends for their endless support. Thank you.
Hi everyone, my name is Emily Hendricks and I'm an undergraduate student. I had the privilege of working in the Medina Bolivar laboratory, and I studied the antioxidant activity of stilbenoid-rich extracts from peanut hairy root cultures. The reason I chose to study the antioxidant activity of these compounds is because we are exposed to oxidative stressors in everyday life, and these stressors cause free radicals to be formed in our body, which are very unstable molecules. And these free radicals can exhibit lots of cell damage that can manifest in diseases all across the body. Antioxidants actually have the ability to donate an electron to the free radical to neutralize them. Antioxidants can be found in a lot of natural food sources. Stilbenoids are phytoalexins, which are inducible plant defense molecules, and they have been shown to have potential applications in human health. They've also been shown to show extensive antioxidant activity. They can be found in peanuts and grapes. And as you can see here, in this picture on the lower left, they are only produced when the plant is under stress. So that yellow mass is the response of the plant producing stilbenoids when attacked by fungus. Resveratrol is by far the most popular stilbenoid. It can be found in blueberries, peanut, and grape. And if you've ever heard that drinking a glass of wine a day can lead to future health benefits, it's because of this molecule here. The peanut produces many stilbenoids, including resveratrol, but what they also produce are prenylated stilbenoids. And as you can see here, these compounds have a very similar structure to resveratrol, but the one thing that differs is this prenyl group. And we hypothesize that this prenyl group can lead to higher bioavailability of these compounds. And what we do to produce these stilbenoids is we use hairy roots as a bioproductive system. What we do is we cut the leaf of a peanut plant and we inoculate it with a bacterium. And when we do that, it produces a disease of the plant known as the hairy root. And these hairy roots are what we use to elicit the stilbenoids. The hypothesis of this experiment was that stilbenoid-rich extracts from peanut hairy roots exhibit significant antioxidant activity. And to study this hypothesis, we are going to elicit the peanut hairy root cultures, extract the phytochemicals, analyze those extracts with HPLC, and assess the antioxidant activity with a combined DPPH-HPLC assay. To elicit the peanut hairy roots, we add the elicitors to 9-day-old peanut hairy roots and let them sit for 168 hours. And as you can see, after elicitation, there is a drastic color change, indicating that stilbenoids are present in the culture media. We extract the stilbenoid extract from this procedure. We use the DPPH assay to determine antioxidant activity. DPPH is a stable free radical, and this assay relies on the antioxidant's ability to reduce the DPPH, and the results are shown colorimetrically. So as you see, when this compound is reduced, a drastic color change occurs, and we can use this color change to calculate the percent inhibition of DPPH. This is the DPPH HPLC assay of that extract. As you can see, the profile that was treated with DPPH shows great inhibition by the arachidin compounds, but no inhibition by the resveratrol. And this is that previous chromatogram quantified. To determine why resveratrol was not inhibiting DPPH in the extract, we fractionated the resveratrol from the extract and performed a DPPH HPLC of that fraction. And as you can see, when isolated, resveratrol readily inhibits DPPH. This indicates that the arachidin compounds and the unknown compounds are better antioxidants in the extract than resveratrol. The conclusions of this experiment are that the composition of the peanut hairy root extract influences the antioxidant activity. The DPPH HPLC assay can screen for unknown antioxidants. The prenylated stilbenoids have higher antioxidant activity than resveratrol in the extract. The extracts have potential pharmaceutical value, and they also have potential value in the food and beverage industry as supplements. I would like to thank Dr. Fabricio Medina Bolivar for letting me work in his laboratory on this project, Patrick Roberto for being my mentor throughout this project, the rest of the Medina Bolivar laboratory for their constant support, and I would like to thank the ABI Biotechnology Research Internship for funding this project. Feel free to leave any questions in the voice thread. Thank you.
Hello everyone, my name is Samuel Whitmire. I am a senior mathematics major here at Arkansas State, and this presentation is over my honors thesis, Fractional Derivatives, Integrals, and the Generalized Fundamental Theorem of Calculus. My mentor, Dr. Meow, and I have been working on this since June 2019. So this thesis covers fractional calculus, which is a subfield of real and complex analysis involving what happens when we take non-integer order derivatives or integrals of functions. For example, in Calculus 1, many students remember the first and second derivative tests, which involve finding the first and second derivatives of a function. Well, we ask what happens when we try to find the 1/2 derivative, or maybe the 2/3, or even an irrational number like the square root of 2 derivative, or integral of a function. The primary goal of this thesis is to unify the proofs of two very popular theorems in standard calculus. The first being the fundamental theorem of calculus, which essentially states that derivatives and integrals are inverse processes, and the second is Taylor's theorem, from which we can approximate a function as its Taylor series of a degree n plus the remainder term defined by, or bounded by, the n plus first derivative of the function on an interval. Section 2 involves Lebesgue measure and absolute continuity. Lebesgue measure is only used to extend our results to a wider scope of functions, because some functions that are not Riemann integrable are Lebesgue integrable. And on the other hand, absolute continuity, which is a type of continuity that is stronger than both uniform and standard continuity, is used primarily in proofs. And we don't have the proofs in this presentation, so we probably will not be seeing absolute continuity much again after this. But it's there for the background information. The gamma and beta functions are also background information that we need. The gamma function is important because it allows us to turn the factorial operator into an operator that applies for any real number, as defined by this integral here. This is important because this is how we define the fractional integral. Beta function, on the other hand, is another part that is mostly used in the proofs, so we probably won't be seeing it again, but it is fundamental for the results that we have in our paper. So to define the fractional integral, we have to start with Cauchy's formula for repeated integration. This is the keystone that the entire fractional calculus operator definition rests on. And this is a formula that allows us to integrate a function n times and see the corresponding output. And the fractional integral takes the factorial operator in the denominator of the Cauchy formula for repeated integration and replaces it with the gamma function, so that we can have a definition, or so that we can apply any real number to that formula. And the fractional derivative is similarly the derivative of a fractional integral. That's how we define the fractional derivative in this paper. Here's some examples of functions for which we have fractional derivatives and integrals. The fractional derivative of a polynomial is not bad, but the fractional integral of a exponential is harrowing to look at, to say the least. Some properties that we like: the integral operators are additive. The derivative of an integral is the original function. Converse is not true. And for the alpha, if derivative of f to be 0, f has to be this polynomial-like term. So section 5 deals with the generalized fundamental theorem of calculus, which this is the theorem itself, but we're mostly interested in the two results that follow. First, that the integral of a derivative for order 1 is equal to f of x minus f of a, similarly to fundamental theorem of calculus part 2. And second, that that a function can be represented as its Taylor series using this generalized fundamental theorem of calculus for a natural number n. The reason being is that if we move this term to the other side, we will see that f of x equals its Taylor series plus its remainder term. Section 6 deals with Abel's integral equation, an equation developed by Niels Henrik Abel to solve a physical problem in the 1800s. This is our definition of it. And the solution for 0 less than alpha less than 1 is given by the derivative of this statement. We use standard calculus to prove this. For the general term for any alpha in real numbers, we again have a solution that involves fractional calculus using the alpha derivative of f. So that is all that we did in this thesis. I hope that you enjoyed watching this. Please stay safe and have a good remainder of the day.
Hello everyone, my name is Christian Rodell. Today I'm going to be sharing with you the research that myself and colleague Ilden Ahmed have been working on this semester in the Arkansas Biosciences Institute regarding the optimization of culture media conditions in peanut hairy root cultures. Our mentor is Dr. Fabricio Medina Bolivar of the Biology Department at Arkansas State University. Here you can see the poster we made. Some of it may be hard to see or read, but don't worry, we will be guiding you through it. Oxidative stress is caused by the presence of free radicals. Free radicals can cause harm on a cellular level by carrying out unwanted reactions. This can eventually lead to larger problems in diseases such as Alzheimer's, cardiovascular disease, and even cancer. Luckily, we have compounds called antioxidants to combat the effects of free radicals within our bodies. One of the most prominent antioxidants on the market today is resveratrol, a stilbenoid. It is found in a variety of plants including grapes, blueberries, and peanuts. The problem with resveratrol as a supplement is its low bioavailability within the human body. Our lab has found derivatives of resveratrol in peanut hairy root cultures that could potentially have a higher bioavailability than that of resveratrol. This belief is due to the presence of the prenylation of the compound, which you can see highlighted on your screen. The peanut hairy root cultures are made using an agrobacterium transformation, which inserts the plasmid into the genome of the plant. The integration of the plasmid allows root tips from an inoculation site to be grown in media with sufficient nutrients. The hairy root system is perfect for the biosynthesis of human supplements due to its high renewability and sustainability. Our primary objectives within this study were to look at factors that could have a possible increase in root biomass, in hopes of also increasing or changing the stilbenoid profile. These variables include the concentration of sucrose, the volume of media present within a flask, and the volume of the flask itself. Our hypothesis is that the increase in growth parameters such as these will be directly related to an increase in root biomass. Isildin will be taking you guys through the rest of the PowerPoint, so I hope you guys have a wonderful week and stay safe.
Thank you, Kristen. Hello, everybody. My name is Isildin Ahmed. I'm an undergraduate biology student here at Arkansas State University, and today I'll be walking you through our experiments, our results, our overall conclusions, and what the future holds for our experiments. So the first experiment we have here is increasing sugar concentration on hairy roots, and what we have is 4 different groups. The first group is 30 grams per liter, which is our current procedure on control, 40 grams per liter, 50 grams per liter, and 60 grams per liter. What we did is we placed 12 tips of hairy roots into the cultures for 14 days, let them grow, and then we froze them for 3 days and lyophilized them in the lyophilizer for 3 days. And this experimental design is consistent throughout our 3 different experiments, with the only difference being the variables. So as you can see from the results, increasing the sugar concentration has had an adverse effect on the root biomass, except with the 40 grams per liter, which showed our best results because it has the most biomass. Our second experiment is increasing the media volume, and with this, what we basically did is we doubled the growth media from 50 mils, which is our current procedure, to 100 mils. As you can see from the results, that increase also translated into increasing the root biomass, and the pictures also translate that too. Our third experiment is increasing the flask and media volume, and what we did here is we increased the flask volume from 250 mils to 500 mils of volume, and the media volume from 50 mils to 100 mils and 200 mils respectively. And the results from those experiments, as you can see, is increasing the flask and media volume has shown tremendous effect in that it increased the root biomass tremendously. And our growth index here shows you how where the roots started off from 12 tips and how they grew in the 14-day period. Our conclusions is that our hypothesis is confirmed. Increasing the growth parameters increases the root biomass. The only limitation to that being the sugar concentration, and that increasing sugar concentration does not increase root biomass except with the 40 grams per liter. And the group that showed the best results, the most biomass, is our 500 ml flasks with the 100 ml media volume. And our future work will be with elicitation. We'll be eliciting the best groups as well as the 40 grams per liter. And we'll be working closer with the sugar concentrations to see what's the most optimum concentration. We'll be doing HPLC analysis to see if the silvinoid profile that we have has been altered by changing the growth parameters, and repeating these experiments in a large scale to find out how consistent our results are. I would like to thank Dr. Fabricio Medina Bolivar for giving us the opportunity to work in his lab, Patrick Roberto and other lab members, ABI, and Create A-State. And lastly, I would like to thank you for giving us some of your time, and have a great day. Thank you so much.
Hi, my name is Grant Dawson. I'm an undergraduate in the Newman Lee Lab, mentored by Lindsay Martin Krikorian, and today I'm asking the question, are the turtles stressed too? Just like you and I, turtles are able to experience stress, not by impending deadlines, but by parasite loads, predator-prey interactions, and other stressors in the wild. When stressed, turtles will release glucocorticoids, a class of bloodstream molecules that have an effect throughout the entire body, specifically to promote the mobilization of sugars to use to respond to the stress and mediate physiological trade-offs. They also shift their immune system resources, changing the relative amount of heterophils and lymphocytes, two types of white blood cells, in their circulatory system. By increasing the relative amount of heterophils, turtles are able to bolster their frontline immune defenses to fight off the possible source of their stress. In reptiles, we find that these responses to acute or intermediate stress are very individualistic, dependent on how much chronic or continual stress the organism is usually under. A fairly unstressed individual can mount a robust stress response, while a chronically stressed turtle will generate relatively weak stress responses due to energy limitations. This is known as allostasis, or adjusting to a new normal. In this study, I wanted to determine if a population of turtles is experiencing chronic stress due to the water quality of their habitat, such as temperature, pH, conductivity, and dissolved oxygen. I measured the blood glucose level of turtles as a proxy for the amount of glucocorticoids released. Blood was taken and analyzed to establish a heterophil lymphocyte ratio. This is both indicative of chronic stress in wild animals as well as shifting immune resources. Together, these metrics will indicate how robustly the turtle is responding to the stress of being caught by researchers, which we can use to infer how much chronic stress the turtle is experiencing. Over 2 weekends, I sampled turtles with the Newman Lee Lab from 3 locations at a runoff site of the Languille River near Cherry Valley, Arkansas. 12 turtles were caught using a hoop net, and their blood was taken from the caudal vein 3 times, 0, 30, and 60 minutes after capture. The blood was tested with a glucometer and smeared onto slides at each of these intervals. The slides were fixed in methanol, stained, and analyzed under a microscope for heterophil and lymphocyte frequencies per 50 white blood cells. Water temperature, pH, conductivity, and dissolved oxygen were measured after each capture using a handheld calibrated device. The results show no correlation between overall change in blood glucose level and the water quality. However, there were positive correlations between blood glucose level and pH and temperature. Heterophil lymphocyte ratios and blood glucose levels both generally increased over time. Figure 3 shows a surprisingly tight correlation between the pH of the water and the baseline blood glucose levels. This is a novel discovery that isn't easily found in the literature, though we hypothesize that it could be due to physiological or ecological factors. Turtles have extremely flexible blood pH levels, which allows them to increase or decrease metabolic production. The level of pH in the water may help to increase metabolic rate. The pH could also be a reflection of limnological factors such as primary productivity or eutrophication. In figure 4, the positive correlation between baseline blood glucose and temperature is due to the ectothermic nature of reptiles. This means that they cannot create body heat, so they must utilize outside sources, which dictates bodily functions like movement and metabolism. As the ambient temperature increases, the turtle's metabolism increases, causing the amount of glucose in the blood to increase as well. Figure 5 shows that heterophil lymphocyte ratios increased over time after capture by an average of 5 heterophils per lymphocyte. Each line on this figure represents an individual turtle, illustrating that as the turtles experienced stress by being caught by researchers, they began increasing their populations of heterophils. This figure shows that some individuals have a smaller increase in ratio over time than others. These turtles with a smaller change in ratio are likely chronically stressed. Therefore, they have less energy to devote to shifting their heterophil lymphocyte ratios. The individuals with a much greater increase likely have enough energy to do so because they're relatively unstressed. With a variation of increases in ratio, we can assume that this sample of turtles is variably stressed. Figure 6 shows all blood glucose levels increased over time by an average of 42 milligrams per deciliter. The uptick in glucose is caused by a host of physiological factors such as the release of glucocorticoids. According to Newman Lee et al. 2019, glucocorticoids aren't as connected to blood glucose levels as once thought. However, the combined efforts of glucocorticoids and other compounds, such as norepinephrine, provide a considerable link between stress and blood glucose levels. Therefore, we can conclude that all these individuals experienced acute stress upon capture. This turtle likely has an artificially high blood glucose level due to having a recent meal. Recent meals could also affect the relationship between blood glucose and temperature, which we need to control for a variable in future experiments. In summary, with these data, we can conclude that the turtles are in fact stressed too. However, this probably isn't from the water quality metrics we tested, and further studies are necessary to consider other variables, such as climate change and anthropogenic influence. Thank you to Mason Fisher and the Newman Lee Lab for their help with data collection. Thank you to Jen Terry for allowing me to conduct this study parallel to her thesis, and thank you to ABI for funding this project.
Good day to you all. My name is Janet Nilmik and I'm here to talk about my project, which is studying transcription factor activity in soybean using the root hair system. And my mentor for this project is Dr. Asela Vijayaratnam. So here is the post I made for my project, and I'll be breaking down each section in the following slides. So the reason why we do this is there is a very prominent disease caused by a pathogen on Phytophthora zoygae in soybeans, and there's an annual loss of $20 million of crop loss in the US alone. Normally there is a defense mechanism inside the soybean plants against P. zoygae that is activated by RPS proteins and RPS genes. However, with time, P. zoygae has developed a resistance to this mechanism, but we have identified critical regulated genes using co-expression network analysis. So therefore, my project is to find the functions of these genes using CRISPR-Cas9 technology embedded into the root hair system. So first of all, what is the CRISPR-Cas9 system? It contains two important molecules, which is the guide RNA, which is a predesigned RNA sequence, and then an enzyme called Cas9. So the guide RNA guides the Cas9 to the target location, and the guide RNA attaches to the DNA, and the enzyme is able to cleave it at specific locations. And once the DNA is being repaired, they form a mutation. The hair root system is mainly due to a plant infecting pathogen known as Agrobacterium, and the strain we use is Agrobacterium rhizogens. And they form hair roots because of a special plasmid known as the TI plasmid, and we can use any of these explants for the infection. So the first part of this project is to develop the CRISPR-Cas9 constructs. So we designed these three protospaces, and using the entry vector, we digested both the entry vector and the destination vector using restriction enzymes. And we are able to ligate the gRNA and the protospace into the destination vector, which is this step. And then after ligating, we transformed it into agrobacteria, and finally we should inoculate it into germinated seedlings. So for this, I used the cotyledon part of the germinated seedling. So the result, the figure 3 shows the digestion of the shuttle vector and the binder vector. As you can see, the undigested vector has a size around 3,500 base pairs, and it has many bands because it has different conformations. And the undigested vector has a size of 500 base pairs, which shows the digestion was successful. Figure 4 shows the screening of the destination vector after the ligation. and there are positive band. For it to be positive, there should be 2 bands in the sizes of 326 base pairs and 296 base pairs. So the blue arrow shows the positive colonies for the 33-34 protospacer, while the red arrows show the positive colonies for the 29 and 30. So we took those positive colonies and sent the samples for sequencing, and figure 5 shows the alignments done between the sequences. Because both the alignments have a high similarity, we decided that we can move forward with the experiment. So the conclusion is that I was able to construct 3 CRISPR-Cas9 products with different protospaces. I was also able to transform them into Agrobacterium rhizogenous K599 strain so that I can store them in glycerol stocks and inoculate them. I did attempt to generate hairy roots 2 times, but both attempts were not successful. So the future approaches for my project is to continue inoculating germinated seedlings, especially the cotyledon, so that I can transform into hairy roots. So once I get the transgenic roots, I should be able to do a PCR to confirm the mutations of the CRISPR-Cas9 technology. I was not able to continue this because of what's happening right now. And these are the references I use. And I also want to thank the research that is funded by my grant, as well as Dr. Fabrizio Medina Bolívar and my mentor, Dr. Acela, and all my lab mates who helped me in my project. And I would especially like to thank the ABI for giving me this opportunity. Thank you.
Hello, my name is Lin-Chi Ho. I am currently a junior biology pre-med major, and I work at the Arkansas Biosciences Institute in Dr. Fabricio Medina Bolivar's lab. I do research with another undergraduate student named Cecily Long and a doctoral student named Zebedee Mohammed Hosenpour. The title of my poster presentation is Evaluating Prenulated Stilbenoids from Peanut as Potential Therapeutic Agents for Triple-Negative Breast Cancer. This is a full snapshot of my poster, and within the next few minutes, I will break it down into its smaller components so that you can get a better glimpse of it. Everyone in some way knows someone who is impacted by cancer. It's such a common disease nowadays, and breast cancer is one of the most common types of cancer in women worldwide. One of the deadliest types of breast cancer is triple-negative breast cancer, or TNBC. The triple-negative means that it is lacking in 3 receptors that most anti-cancer medicines target in order to stop the spread of cancer. And because of this, TNBC has very few treatment options. When plants, such as peanut, are stressed, they produce these defensive compounds called prenulated stilbenoids. And we have found that these stilbenoids have properties such as anti-cancer, antioxidant, anti-inflammatory, and more. The focus of this research, however, is to look at the anti-cancer property of these prenulated stilbenoids. My hypothesis is that the prenulated stilbenoids arachidin-1 and arachidin-3 will induce more apoptosis in TNBC cells than the non-prenulated stilbenoid resveratrol. Apoptosis is essentially the death of cells. In this research, my objective is to compare the apoptotic effects of arachidin-1, arachidin-3, and resveratrol on TNBC cells using flow cytometry. Flow cytometry is a technique that helps to detect some characteristics of cells. Future studies conducted would use different assays or tests to gain more results. In order to produce the stilbenoids for testing, I treated peanut hairy roots with 4 different elicitors or chemicals to stress the roots and make them produce their defensive compounds. Hairy roots are an immortalized plant system, so they will stay as hairy roots forever and will produce the same compounds as plants do. Once the stilbenoids are produced, they are separated using semi-preparative high-performance liquid chromatography as shown here, and then we can choose our stilbenoids for testing. Arachidin-1 and arachidin-3 are called the prenylated stilbenoids due to this extra tail added on that resveratrol does not have. The stilbenoids are then added to the TNBC cells in different concentrations, and then I can run tests on them. Using a flow cytometry assay, the number of cancer cells in each stage of mitosis can be seen. 3 days after treating the cells with the compounds, arachidin-1 and arachidin-3 have more cells doing apoptosis or are in the G0 phase where the cells are not growing compared to those cells treated with resveratrol. At lower concentrations of these compounds, this pattern is more easily seen. From my results, I can conclude that arachidin-1 and arachidin-3 at lower concentrations induce more apoptosis in cancer cells compared to resveratrol. The reason for this is the extraperennial tail that arachidin-1 and arachidin-3 have, but that resveratrol doesn't. This small extra piece results in a higher anti-cancer property. This poster was presented at the Create at State Symposium, and my research is supported by the USDA and the Arkansas Biosciences Institute at Arkansas State University.
A very good day to you all. My name is Angela Gigi and I'm from Department of Biological Sciences, Arkansas State University. And my mentor is Dr. Asela, and today I'm going to talk about assessing the performance of lightweight aligner for plant transcriptome. As technology has been advancing day by day, methods of analyzing data has also been improved. Traditional analysis, which takes hours to complete, can be done within minutes using these newer platforms. But this analysis has only been performed in humans and other model animals, but has not been done in plants. So my goal is to use platforms such as RStudios to compare the results between traditional and novel technology. And if the results are similar, then these newer softwares can be used in further analysis, which can save a lot of computational time. RNA sequencing is a technology where we extract RNA from the cells, fragment this RNA, convert it into cDNA, and then send it for sequencing. This sequence can be further used for aligning short read to references, count the number of short read aligned, and then do a differential gene expression analysis. One of the alignment tool that I'm going to use for this experiment is called Bowtie2. Bowtie2 is a memory efficient tool for aligning and sequencing. One of the main benefits of Bowtie is that it is more tolerant to low quality bases. But as you can see on your right, the time used for Bowtie analysis is too much compared to all the other softwares. The second alignment tool I'll be using is Kallisto. Kallisto is a mapping program. It creates some sort of approximation of the reference and read rather than actually matching it to the sequence. As you can see, this is a comparison between time and accuracy. In the first graph, we can see the time used by Kallisto and time used by Bowtie has a huge difference. Where Kallisto just uses 5 minutes, Bowtie takes almost 1000 minutes to do the same sequencing. And when we are comparing the accuracy, we can see Kallisto and Bowtie have almost the same number in accuracy. Why are we doing this in plants? Plants have a large variation in their genome size and their level of ploidy. This diverse and complex nature of plants make their genome very difficult to analyze. And callisto has never been tested in polyploidy plants, but have recently been done with diploid plants. So a very systematic study of this genome is important, and these tools are important so that we can save time and other resources in future. The goal of my project is to use diploid crop plant samples to compare different alignment methods to reduce the analysis time, and to develop an analytical pipeline for this crop for future references. And the main objective is to compare the performance of callisto and bowtie 2 in Arabidopsis sample. The data for this experiment was obtained from a paper which originally had 17 samples, but due to time restraints, we could only do 10 samples. The methods I will be using is use the reference and map it using callisto and bowtie 2, and then do an abundant estimate using callisto and rsim, and then do a differential gene expression and assess performance using RStudios. After analyzing both the softwares, we got a volcano plot and a PCA plot. As you can see in figure 8a and 8c, both the plots look exactly similar with very, very minute changes, as in the case of 8b and 8d. We can see all the groups were almost group similar. So from this plot, we can see that there is no much difference between the use of callisto and bowtie 2. From this Venn diagram, we can see that majority of the area has been overlapped between the differentially expressed gene. There is a very slight difference or very slight area which there is no overlap, and it can be majorly because of some false positive because they are from the same experiment. This is a plot showing the count between bowtie 2 and callisto. As you can see, the red one represents the bowtie and the blue one represents the callisto. And we can see the difference remain consistent throughout. I think callisto stands out because the repetitions may have been counted while bowtie didn't count it. From the results, we can see that there is no much difference between callisto and bowtie's analysis. But when we did this experiment, callisto took just 3 hours, where bowtie took more than 2 days. So we can conclude that callisto is ultra fast and almost gave us same accurate result as bowtie. And in future, this lightweight and fewer options will make the everyday analysis so much more easier. And we can see from the initial analysis that it can be used in non-human or mouse samples. And callisto and all the other newer technologies can change the way we do data analysis. And these are the citations of the paper that I used for this project. Thank you and have a great day.
Hello, my name is Rebecca Martin, and I am a student researcher in the Hirschberger Laboratory in the Department of Chemistry and Physics at Arkansas State University. Today, I will be presenting a method of synthesis for new 2-aminopyrimidines. With our current global situation, the development of new medicines to treat current and novel diseases is of the utmost importance. The list of underlying conditions that can make the COVID-19 virus lethal is long, and not all of those conditions are quickly treated. My research works to develop new molecules that may have relevant activities in developing new medicines through an efficient synthesis. The products utilize an organic compound found in many different types of existing drugs, called a 2-aminopyrimidine group. This structure is the basis of medicines that treat HIV, cancer, thrombosis, and many other diseases. A few of these can be seen under the medicinal 2-aminopyrimidines section, with the 2-aminopyrimidines group highlighted in red. The first step in the reaction scheme is the synthesis of a bigonelli compound using an aryl aldehyde, urea, and methyl acetoacetate, as shown in Figure 1. The product can be varied depending on the aryl aldehyde used. For example, in my research, I used compounds with halide atoms, like bromine and chlorine, attached to the benzene ring in the aldehyde. These atoms appear in the bigonelli product and change some of its chemical and physical properties. The second step is the oxidation of the bigonelli product, as shown in Figure 2. In this reaction, 2 hydrogen atoms are removed from the compound, and the structure isomerizes to the alcohol conformation. This occurs because of its increased stability over the ketone conformation that is an intermediate in the reaction. The final step is an Ugi-Smiles reaction using the oxidized bigonelli product, seen in Figure 3. The other reagents are an aldehyde, a primary amine, and an isocyanide. The specific aldehyde and amine used can also vary the final product of this reaction that contains the desired 2-aminopyrimidine group. Aliphatic aldehydes were used after discovering that aromatic aldehydes did not allow the reaction to go to completion and create the desired product. All of the compounds I produced using this multi-step synthesis were structurally verified using nuclear magnetic resonance spectroscopy. One of the spectra created and the product it represents is shown in Figure 4. The purification and isolation of the products are still being completed using thin layer chromatography and column chromatography. Once these steps are completed for all of the compounds produced, biological testing, such as enzyme assays, will occur to determine their likelihood of being used in new pharmaceuticals. I would like to thank the Department of Chemistry and Physics and the College of Science and Mathematics at Arkansas State, as well as the FRAC, for their support of this work. Thank you.
Hello, my name is Sean King, and I am an undergraduate student at Arkansas State University. I am a biological science major, and I had the pleasure of participating in the Arkansas Biosciences Institute internship in the summer of 2019 with Dr. Newman Lee and her lab. I have continued to work in the lab, my focus being on my work to examine blood samples from radiated sliders by making smears and observing them under the microscope for immune cell morphology and relative proportions. Red-eared sliders are turtles that are common in northeastern Arkansas. They are found in many habitats, but we examine them in water surrounding agricultural farmlands. In the wild, turtles are exposed to many kinds of stress and infections such as endoparasites. One endoparasite is the hemoparasite Plasmodium. These blood parasites are visible in blood smears. Mosquitoes are their primary vector. In addition to examining parasites, blood smears allow us to examine important immune system cells, or white blood cells, such as eosinophils, which are high in times of parasite infection, heterophils, which are phagocytic cells, and lymphocytes, which are a second line of defense in their cells. Turtle immunology is not very well known, so studying basic cell biology has huge benefits to understanding disease and immune function in turtles. The main goal of this project was to get a baseline immune cell proportions for red-eared sliders by determining their heterophil lymphocyte ratio, also called HL, or HLR in my poster, eosinophil count, and measuring Plasmodium occurrence. Blood samples were collected at intervals of baseline, 15 minutes and 30 minutes from the caudal vein and the tail in the summer months of 2019 in Beritage Farms, Cherry Valley, Arkansas. Blood film slides were taken and smeared, then fixed in methanol. Smears fixed were then taken to the lab to be stained in James Wright stain for 1 minute stain, 1 minute buffer, and 10 dips of rinse. Slides were then dried overnight. Slides were examined under the Nikon 1200 light microscope for observation of red blood cells and white blood cells. I noted any plasmodium in the red blood cells and counted eosinophils, heterophils, and lymphocytes. I would like to refer your attention to the figure on the right. The x-axis has heterophil lymphocyte ratio, and the y-axis has the number of plasmodium. There was no relationship between plasmodium and HL ratio in the radiated sliders sampled. The graph in the middle, the x-axis has the number of eosinophils graphed, and the y-axis has the number of plasmodium. We found no relationship between plasmodium and eosinophil counts. The graph on the left, the x-axis is sex, the y-axis is HL ratio. We found no difference in HL ratio between male and female turtles. HL ratio is typically indicative of higher stress, so we expected to find a difference between the female and male HL ratio because female turtles expend energy on egg production during the mating season, which has during the sampling period we took. We also expected that all turtles would have a higher HL ratio and a higher plasmodium load. We did not see either of these relationships. It is possible that other infections could have been present. There are other stressors that we did not measure. We also predicted a higher level of eosinophils in turtles with higher levels of plasmodium. In mammals, eosinophils function as antiparasitic cells, but their role may be different in reptiles. Additionally, different seasons could influence plasmodium occurrence since mosquitoes are more active during the summer months, which was when the samples were collected. Finally, we don't know what these turtles have experienced prior to capture. The environment of other stressors and infections could have influenced the results when blood was sampled. Future work could be determining plasmodium species present and increasing the sample size to include more individuals. With that, I am very thankful to my mentors, Dr. Newman Lee and Benjamin Spitz. I am also very grateful to the help from the members of the Newman Lee Lab. Finally, I am so grateful for the opportunity to do this work through the ABI internship at Arkansas State University.
Hello, my name is Jessica Parr. I am a student at Arkansas State University, and I work in Dr. Ali's research laboratory in the Chemistry and Physics department. My presentation is about the absorption of catechol and hydroquinone on the surface of titanium dioxide. Currently, many people are talking about the environmental changes and issues happening to our planet. You hear about global warming and air pollution, but water pollution is not mentioned as much. Water pollution is a huge part of the environmental changes that need to be improved. We are using products that go down our drain into our water, such as soaps, shampoos, and hair dyes. These products are known as personal care products, or PCPs. PCPs have newly made chemicals in them that our water plants are not able to fully remove due to their old age. Therefore, these compounds are not being able to filter out of our water, and they start to build up in our water cycle. These compounds can cause harm to us. In my research, I look at two chemicals that are present in PCPs. Those chemicals are catechol and hydroquinone. Catechol is a chemical found in hair dyes. When this compound comes in contact with skin, it causes severe skin burns. Hydroquinone is found in the resin coating in hands. It causes severe eye damage and genetic defects. The Ali Laboratory at Arkansas State University looks at novel ways to remove harmful chemicals from the water. In the case of my project, we would like for these chemicals to be removed from our water, especially during this time with COVID-19, a new disease for which we need constant access to clean water to keep us healthy. With us washing our hands even more, it is important to continue having clean water that is safe. In my research, titanium dioxide was used as a filter to trap these chemicals on its surface, so that these harmful chemicals can then be removed from the water. To investigate the ability of titanium dioxide as a filter, we use 3 different metrics of adsorption: isotherm, kinetic, and pH absorption. When observing its isotherm absorption ability at different concentrations, each concentration was absorbed after a 24-hour period. It was found that as the concentrations increased, so does the absorption of the chemicals to titanium dioxide, as you can see in Figure 3. The trend that was found is that as the concentration of these chemicals increase, the more likely the chemicals will attach to the surface of the titanium dioxide. When examining the reactions at the kinetic absorption over different times, it was found that at 20 minutes, that most of the chemicals were absorbed to the surface of the titanium dioxide. This can be seen in Figure 4. As time continues, no more of the chemicals were removed. Then looking at pH absorption on Figure 5 of both catechol and hydroquinone, reaching the peak absorption at around pH 8. After pH 8, the absorption starts to decrease. This makes it easier when designing a filter because water's pH is usually around pH 7. Therefore, the pH will not have to be changed in order for the chemicals to be mostly absorbed to the titanium dioxide. Now, going back to the isotherm absorption and the kinetic absorption, hydroquinone is absorbed more than catechol. After looking into why this is happening, this is because hydroquinone and catechol have the same molecular formula but different structures. Both structures for these chemicals have two OH groups, which can be seen on figures 1A and 1B. The OH group for hydroquinone is in the pair position, while the OH group for catechol is in the ortho position. Therefore, it is easier for hydroquinone to attach to the titanium dioxide structure because it has less steric hindrance. The catechol structure has more steric hindrance because of the two OH groups being next to each other. In the future, we can use this information to design a titanium dioxide-based filter to remove hydroquinone and catechol from our water. Also, to be able to see if titanium dioxide can be used as a filter for other harmful chemicals.
Hello, my name is Noah Smith and I work at Arkansas State University in Dr. Hirschberger's lab. Today I am here to talk about a novel method to potentially develop medically useful compounds. It is simple, easy, efficient, and uses well-established reactions in a new way. The method creates chemicals that contain the 2-amino pyrimidine group, a functional group present in several antifungal, antibacterial, and anti-cancer drugs. Meaning it is useful to create new 2-amino pyrimidine containing chemicals as they have a chance of being medically viable. First, I want to talk about the reaction series devised for this purpose. This can be seen in the section labeled Methods at the bottom left of the poster. This reaction series is 3 steps: a Bigonelli reaction, an oxidation, and Ugi-Smiles reaction. These reactions are well studied, easy, and efficient. The novel portion of this design is that they are performed in series and that a Bigonelli product has not been used in an Ugi-Smiles reaction before. The results of this method can be seen in the Results section in the middle of the poster. The columns of this table are, from left to right, the reaction ID, the amine used in the Ugi-Smiles reaction, the aldehyde used in the Ugi-Smiles reaction, the physical structure of the specific product, and the yield of the reaction. As can be seen in the yield column, the yields vary from 20% to 70%. So the reaction series can be said to have a moderate yield. Next is a section labeled Purification at the top right. This concept is important as it removes any unwanted products from the desired products. This allows structural conformation by NMR and IR. It works by passing a mobile phase of a certain polarity in the crude product through the stationary phase of a different polarity. This causes all the chemicals present in the crude product to separate based on their polarities, as the individual compounds will stick to the stationary phase at different rates, making it possible to remove all unwanted chemical products. Again, this is very useful in all areas of chemistry and pharmaceuticals to prevent unwanted effects and reactions. The pure produced chemicals' biological effects are listed in the section directly below the previous. They were tested by the use of a disk diffusion assay, which tests the size of inhibition zones created by the experimental products versus a known antibiotic. Several different bacteria were tested against, including both gram-positive and gram-negative bacteria. The results, as listed, were that all products failed to produce an inhibition zone, meaning they failed to produce any inhibitory effects on the tested bacteria. So, all in all, this experiment succeeded in producing the expected structures, had relatively good yields, but failed to produce any effects on the bacteria. This result is acceptable as the purpose of this experiment was to devise a method to produce various 2-amino pyrimidine containing chemicals, which it did. Just because the produced chemicals do not have any effects on the bacteria does not mean all the chemicals this method is able to produce are ineffective. In the future, more work will need to be done to further increase the yields of the reaction and test a wider range of bacteria, insects, and fungus. I would like to thank Dr. Alam for allowing the use of his equipment, the FRAC for funding, and Hansaraj and Grace Hawk for help performing the biological test. Thank you.
Hello everyone, my name is Nathan Stevens. I am a senior at Brooklyn High School. Today I will be presenting a poster for the project that I am currently involved in. I got the opportunity to be involved in this project work under my mentors Sharon Padul and Dr. Asila through the Young Exceptional Scholars Program. The topic for my poster presentation is mapping transcription factor interaction in the soybean. The soybean is an important crop for the U.S. It carries rich nutritional and economical value. By studying transcription factors and their interaction with each other helps us to understand the gene expression of the proteome in a soybean. The study of their transcription factors is very important because it allows us to generate superior plants in the sense of defensive productivity. We are focusing our project on defense-regulated transcription factors and their interaction during gene regulation. In Figure 1, we can see that the transcription factor binds the binding site and helps the gene to regulate, eventually to produce a different protein. In Figure 2, you can see how two different proteins are interacting with each other to bind to the DNA to express that gene. And in Figure 3, demonstrates how molecular cloning works. This figure shows how we can insert the gene of interest into the vector, which will transport the gene of interest to the competent cell and multiply vectors when cultured. Vectors are DNA plasmids found in bacteria. The goal for this project is to establish a protocol to study the protein-protein interaction between defense-regulated transcription factors and our interest. Specifically, our objectives for this project are to clone genes into GST and halo-tagged vectors and define 8 protein-protein interactions between these transcription factors. We began by extracting 8 open reading frames from our soybean, and then it was cloned into our donor vector using BP cloning method. Now we call this donor vector an entry clone because it has our gene of interest. These entry clones were then transformed into E. coli and were cultured in LB plus kanamycin media. Since this vector is resistant to the kanamycin, it will allow cells to grow in this media. After the incubation of the media, colonies of PCR was done to detect the clone vector and gel electrophoresis. After this step, plasmid extraction was done from the positive colonies. The inserted gene in the entry clone was then transferred to the destination vector using LR cloning. Since the vector is resistant to carboxylin, they were cultured in LB plus carboxylin media to ensure growth of the cell correctly inserted with the destination vector. After colony PCR and purification, we sent these plasmids for sequencing. After getting our results, we compared our sequences with a database using BLAST. This is our entry vector that we used. This is our destination vector that we sequenced with our genes of interest. In figure 4, we can see the results of our gel electrophoresis. This showed us that the colonies were good for the sequencing. In figures 5 and 6, we can see the BLAST results that showed us the GST tag and open reading frame were inserted into our vector and were aligned with good score. Looking into these results, we could conclude that the open reading frames were cloned, and in the vector, the results were good. In the future, we are aiming to detect transcription factor interaction using the wheat germ extract, followed by the pull-down assay for our final detection. In this step, we will be tagging our protein with both GST and halo tags. Finally, I want to thank 2020 Young Exceptional Scholar Program for this opportunity and the funding, Arkansas State University and Arkansas Biosciences Institute for the facilities and support. Thank you everyone for listening to my oral poster presentation. I hope you have a nice day.
Good day everyone. I am Aisha Siddiqa. My faculty advisor is Dr. Alum from chemistry. The title of my presentation is Cytotoxic Effects of Cancer Compound on Human Embryonic Kidney Cells. The second slide shows the details of my poster presentation, which I am going to talk next. Let me give you some background and significance of my study. Cytotoxicity is a widely used indicator for determining the effectiveness of chemical compounds, and cell-based assays are often used for screening the compounds. It is important to know how many viable cells remains at the end of the experiment so the proper compound and their dose level can be determined. The main objective of my study is to assess cytotoxic effects of selected active compounds developed in Dr. Alum's lab and their concentration on human embryonic kidney cells. Some researchers around the globe perform similar studies with different cells and compounds. For example, Prasad et al. Studied several novel N10 substituted acridone derivatives bearing alkaline side chain with tertiary amine group against hex cells. Hamilton et al. Studied cytotoxic effect of fascaplycin against small lung cancer cell line and found it to be suitable for treatment based on high cytotoxic activity. In terms of materials, we consider 4 compounds with different concentrations and tamoxifen as the control drug. We used HEC 2093 cell line. The compounds are antibiotics A4 bromo coumarin and A31 CF3 coumarin with concentration of 25 and 50 microgram per ml, and anti-cancer compound R21 methoxythiazole ethisterone and R23 pyridine thiazole ethisterone with a concentration from 100 to 3.125 microgram per ml. Our experimental setup was in vitro cytotoxicity that consisted of 4 major steps: plating cells on day 1, adding compound on day 2, risazurin assay on day 3, which was followed by analysis on the same day. The analyses are done by using a citation 5 cell imaging reader. Now results. The average and standard deviation of 3 triplets are shown in the bar charts. For antibiotics, A31 is less toxic than A4. The higher concentration of A31 is less toxic. A31 is even better than tamoxifen, but A4 had a complete toxicity. For antimelanoma compounds based on the half maximal inhibitory concentration, R21 is more toxic than R23. Based on the experimental result, we conclude that results are promising. Chemical compound A31 with a concentration of 50 microgram per ml is better. Between anti-melanoma compounds, R23 is a better choice. However, more cells to be tested to support findings. I would like to thank A-State, ABI, ORTT for giving me the opportunity to conduct the study and deliver this presentation. Also, thanks to my advisor Dr. Alom and Mr. Stephen Chambers for helping me with conduct these experiments. I have provided details of cited reference, Prasad et al. And Hamilton et al. Thank you all and have a wonderful day.
Hi, I'm Rokib Hassan, a PhD candidate in molecular bioscience PhD program here at A-State, and my advisor is Dr. Fabricio Medina Bolivar. Today, I'm presenting my research on finding the anti-inflammatory mechanism of arachidin 3. Around 40% of the hospitalized coronavirus patients develop ARDS, which is caused by lung inflammation due to the virus infection. Because of the inflammation, fluid and immune cells are going inside the lung alveoli, resulting interruption of gas exchange, which causing breathing problem, and patients needed ventilation to breathe. Unfortunately, almost 50% of those ARDS patients are dying based on the current research. So lung inflammation is causing death in this case. Chronic inflammation can also cause and help to progress many other diseases like heart disease, cancer, Alzheimer's, etc. Even though there are few anti-inflammatory drugs available, scientists are constantly looking for more targeted, improved, and less side effect containing drug candidates. If you look closely among the anti-inflammatory drugs here, the latest one is Celecoxib, which was invented in 1995, more than 2 decades ago. Resveratrol, which initially found in red wine and later also found in grape, peanut, and blueberry, showed wide range of health benefits, including anti-inflammatory and anti-cancer activities. However, because of faster metabolism, it has limited bioavailability. Arachidins, which found in peanut, had similar backbone of resveratrol with an extra 5-carbon chain called prenyl group. Collaborative research from our lab found that because of prenyl group, arachidins are potentially more bioavailable, can inhibit virus replication, and can also bind with the cannabinoid receptor. Some literature is also suggesting anti-inflammatory effect of arachidin. However, the exact molecular mechanism of anti-inflammatory effect of arachidin yet to discover. Our lab uses peanut hair root system to produce these arachidins. In this project, we will elicit, extract, and purify the arachidin 3 from the peanut hair root medium and check its anti-inflammatory effect in cell line and rats. Overall goal is to dissect the potential anti-inflammatory role of the arachidin 3, and the hypothesis is that arachidin 3 has better anti-inflammatory activity than resveratrol. After elicitation and extraction of the peanut hair root culture medium, arachidin 3 was purified using HPLC method, which later has been used to treat the cell lines. Cytotoxicity and cell viability has been checked using CellTox screen cytotoxicity and real-time glow empty cell viability assay respectively. Cytotoxicity assay results showing that 5 and 2.5 micromolar concentration is not toxic compared to the vehicle control 0 micromolar, which is the orange line. Similar to cytotoxicity, there is no effect of cell viability in lower concentration of arachidine-3 treatment compared to the control. Later, LPS-induced inflammatory cells were treated with arachidine-3 and found that arachidine-3 significantly reduces the pro-inflammatory cytokine IL-6 production compared to the control. NF-kappa beta signaling pathway plays crucial role in inflammation, and many drugs are specifically targeted for this pathway. Inhibition of any major molecule in this pathway can reduce inflammation. Specifically inhibiting the translocation of the REL-A, which is also known as the P65, can hamper the pro-inflammatory cytokine production. So we did some computational analysis which suggesting that arachidine-3 has a stronger binding affinity for P65 and IKK. To test that, we have used immunofluorescence-based imaging technique and found that arachidine-3 inhibits the translocation of NF-kappa beta P65 complex and also the HMGB1. These results suggesting that arachidine-3 acts as an anti-inflammatory compound by regulating the NF-kappa beta signaling pathway. We have also conducted proteomics from UMS Proteomics Core Facility, where LPS-induced inflammatory cells were treated with arachidines and resveratrol as a positive control. And an animal pilot study on rats have been conducted in collaboration with Dr. Rajagopalan from NYIT to have better understanding of anti-inflammatory functions of arachidines. These experiments are ongoing and data need to be further analyzed. In conclusion, we have found that lower concentration of arachidine-3 is safe and it functions as an anti-inflammatory agent by inhibiting the REL-A complex translocation into the nucleus, which eventually reduces the production of IL-6 and reducing the inflammatory responses. For further experiment, we will use Western blotting and other molecular techniques to check how arachidine-3 affecting other key proteins such as IKK and I-kappa beta in this pathway, and further animal testing will be done for better understanding. Here is the references that I have used. And I want to thank everyone for the research support, including the funding agencies. Thank you so much.
Hello everyone, I'm Uddhav Karki. I'm doing PhD here in Arkansas State University in the Molecular Bioscience Department under the kind supervision of Dr. Jesu. Today I'm going to present part of my PhD dissertation work. In this project, I'm trying to produce cell wall deficient plant cells to enhance the biotherapeutic protein production. I use the tobacco plant and make the callus from this plant, and I try to remove the cell wall, which will enhance the biotherapeutic protein production in the industrial scale. Now moving towards the research background. Those therapeutic materials produced by using the biological means, including change of the DNA, is called biotherapeutics. Human insulin is the first FDA-approved biotherapeutics. It has a great global market and is very costly. More than $1 billion will be needed to make a new drug from concept to the market. Animal and bacterial cell culture are two major production platforms. But still there is some challenges in animal and bacterial cell culture system. Animal cell culture system is costly, time consuming, and there is a possibility of cross contamination. Meanwhile, bacterial system can produce the complex humanized proteins. So plant can be a suitable alternative production platform because it's a cost effective, easy to scale up, safe to use, and can be produced or complex proteins. Protalix Therapeutics is a commercially successful example of plant therapeutics. They produce glucocerebrosidase in the carrot and tobacco cells, which is the first FDA-approved plant-produced therapeutics used for the lysosomal disorder. But still there are some challenges in plant production platforms, like low yields, formation of the big clumps, having the large vacuoles which have the more water, and it makes it difficult for the long-term storage, unlike in animal cells. They have some proteolytic enzyme that can degrade our target proteins. So I think cell wall removal is a promising solution. Now talking about the plant cell anatomy. Plant cell has the cell wall and the large vacuoles, and the cell wall components is made up of the pectin, hemicellulose, and cellulose. They form the interconnecting structure and give the rigidity to the plant cell in the environment, natural environment. Now let's discuss about how modern technology can remove the cell wall permanently. In this system, there is a pre-designed single RNA which leads to the target of our sequence and DNA cutting enzyme called Cas9. They will form the RNA protein complex and transport it to the target DNA sequence in the chromosome. They will make the target gene permanently non-functional. This technology is called CRISPR-Cas9 technology. Now moving towards the research plan. I use two approaches in the research. First one is chemical inhibition approach, and the second one is gene modification approach. At first, I treated the plant cells with a cellulose biosynthesis inhibitor dichlorobenzonitrile and observed the protein secretion, its morphology, and I did the characterization. In the second approach, I used a modern technology which I already mentioned to the target gene knockout. In this system, I used the bacterial system for the transformation of this Cas9 and the guide RNA of my target to the plant cells, and I did the characterization of the transformed cells. Let's talk about result. From the chemical inhibition approach, I found that cells are alive, divide slowly, but formation of the large clumps, which was disappeared after treating the pectin hydrolyzing enzyme pectinase. So these clumps may be due to the presence of large amount of pectin, and there is normal productions and secretion of reporter proteins. What I conclude from this result is that there is no vital role of the cellulose in the plant cell viability in the controlled environmental condition. In the gene modification approach, I successfully knocked out the 115 base pair of cellulose synthase A gene, which is responsible for biosynthesis of cellulose in plant cell wall. But still they are in the mixed form with the wild type. So I'm screening the pure knocked out lines. After selecting the pure knocked out lines, I will characterize its anatomy of the cell wall, and I will check the function of the cells. So I'm targeting the more site of these same genes. That's why I can make it more non-functional. That's why it can lose its functionality permanently. So I can get the cellulose-free plant cell. I'm extremely thankful to my advisor, Dr. Jesu, my committee members, lab members, Arkansas Bioscience Institute and MBS program, Arkansas State University, and all the other funding agencies. Thank you for listening me. Stay safe.
Hello everyone, my name is Sepida Mohammad Hossainpour. I'm a PhD student in Dr. Fabricio Medina Bolivar lab. I want to present my research, which is about using arachidine-1 and arachidine-3 as a potential therapeutical agent for triple-negative breast cancer. Why breast cancer is important? This map shows the mortality of breast cancer globally, from highest mortality in the dark red and to lowest in light red, and USA is in the average of breast cancer mortality. And this map shows the percentage of people who were diagnosed with the breast cancer globally, from highest percentage in dark blue and lowest in the light blue, and USA is in the dark blue area. In 2018, statistics shows more than 266,000 people diagnosed with the breast cancer, which means among 8 women in USA, one of them diagnosed with the breast cancer in their lifetime. For treating breast cancer, we need to understand a term that is called receptor. Receptors are protein on surface of the cells, and they are similar to the doors for the cells. In breast cancer, there are 3 main receptors, which they called estrogen receptor, progesterone receptors, and hormonal epidermal growth factor 2 receptor. The main issue with triple-negative breast cancer is this type of cancer cells lacks of all these 3 main receptors. Why this is the issues? Because when we want to treat the breast cancer by using chemo-targeted or hormonal therapy, we need these receptors, because drug need to enter to the cells through those doors, and triple-negative breast cancer lacks of all these important doors or receptors. In our lab, we are looking for a way to treat breast cancer by using resveratrol and resveratrol derivatives compounds. Resveratrol is a phenolic compound which is extracted from peanut, berries, and grapes. Resveratrol shows high anti-cancer activity, especially on triple-negative breast cancer in vitro studies. But in a clinical study, resveratrol shows liver failure and low bioavailability, and high concentration of resveratrol needed to be effective. So, derivatives of resveratrol have a prenyl groups, which showed in the red and blue circle. We call them arachidine or prenylated stimulants. Arachidine show potentially higher bioavailability in compared to the resveratrol in vitro study. And also, because resveratrol showed anti-cancer activity, we use resveratrol as a control for this study. The hypothesis of this research is prenylated stimulants have higher anti-cancer activity and higher bioavailability in compared to the resveratrol. Arachidine and resveratrol are defense mechanisms for plants. In our lab, we use peanut hair root systems, and we put the peanut hair root system in a stress situation to produce arachidine compounds. Then we extract those compounds by HPLC. After that, we treat 3 different triple-negative cell lines by using arachidine and resveratrol, and then we test those and their functions by using different in vitro assays, like apoptosis assay, flow cytometry, and cytotoxicity assay. Apoptosis assay and cytotoxicity assays results shows the arachidine-1 and arachidine-3, which here you can see arachidine-3 has the higher apoptosis, which is displayed in the red bar, and lower percentage of alive cells by increasing the concentration of arachidine-3 in the blue bars. And also, arachidine-1 showed the higher apoptosis and low percentage of alive cells, or low IC50, in these bar graphs. They represent the better results in compare with the other arachidines and resveratrol. We also showed a similar result by using flow cytometry, which is displayed in this graph, and arachidine-3 in yellow bar and arachidine-1 in green bar. Both shows the good results, especially arachidine-3 showed higher apoptosis in early apoptosis in compared to the control and resveratrol. Therefore, As a conclusion, racketing 1 and 3 shows higher anti-cancer activity in compared with other racketing and resveratrol. And for future study, we want to treat the different type of cell lines, 3 triple negative cell lines and 1 epithelial cell line as a control, and test our racketing 1 and 3 by different in vitro assays to select the best racketing, and then we use that best racketing for in vivo assay. Thanks everyone for listening to my presentation. I want to thank to my professor and all my committee member and my lab members.
My name is Patrick Roberto, and I am a master's student in biology studying under Dr. Fabricio Medina Bolivar. The topic of my presentation is over antioxidants produced naturally in peanut. Antioxidants are immensely important for human health because they prevent the overproduction of free radicals. Free radicals are highly unstable molecules that are linked to inflammation, cancer, heart disease, and neurodegeneration. Therefore, it is recommended to consume a variety of fruits and vegetables that are rich with antioxidants. Interestingly, in peanut, there is a group of antioxidant compounds known as the prenylated stilbenoids that exhibit a chemical modification known as a prenyl unit that may enhance their antioxidant capacity in a cellular environment compared to non-prenylated stilbenoids. In nature, peanut produces the prenylated stilbenoids when they are stressed by the environment. In the lab, it is also possible to stress peanut to produce a high yield of the prenylated stilbenoids using chemical elicitors, methyl jasmonate, cyclodextrin, magnesium chloride, and hydrogen peroxide. By adding these compounds into the culture medium of peanut hairy roots, the hairy roots will secrete the prenylated stilbenoids into the culture medium. The culture medium can then be extracted using organic solvents, and the phytochemical and antioxidant profile can be determined by high-performance liquid chromatography, which separates the compounds based on their polarity. In this example, peanut hairy roots were incubated with the aforementioned elicitors for 210 hours, and resveratrol, arachidin-1, and arachidin-3 were secreted into the culture medium. These three compounds are tremendously antioxidant. And the hypothesis is that an extract that is enriched with several different compounds, including the prenylated stilbenoids, is highly antioxidant in a cellular environment. And to test this hypothesis, it was necessary to develop a cellular antioxidant activity assay based on DCFH-DA staining of HeLa cells. HeLa cells are cancer cells, and they were selected due to their rapid growth. DCFH-DA is capable of crossing the cell membrane where it is rapidly metabolized by the cells, and they are oxidized by free radicals to yield DCF, which is a highly fluorescent product. Therefore, an increase in fluorescence corresponds to an increase in free radical levels. And to test the antioxidant capacity of the extracts in a cellular environment, the following procedure was carefully standardized, and it included seeding 60,000 cells per well and then incubating those cells for 12 hours to 24 hours. After washing the cells carefully with PBS, they were co-treated with 25 micromolar DCFH-DA and the samples at varying concentrations for 1 hour. Then the solution was replaced with hydrogen peroxide for 1 hour, and the fluorescence was measured. And what we found was that treatment of the cells with 100 micromolar hydrogen peroxide resulted in a significant increase in the fluorescence. Co-treatment with 28.5 micrograms per milliliter through 228 micrograms per milliliter produced a significant decrease in the fluorescence, which suggests a significant decrease in free radical levels, which means the extract is highly antioxidant in a cellular environment. To further validate the conclusions of this antioxidant assay, it is necessary to perform cytotoxicity assays to determine if the compounds in the extract are toxic, and then follow up with potential in vivo studies to evaluate their antioxidant activity. And this research would not be possible if it hadn't been for the tremendous support of my advisor and my lab members. And I would also like to thank the Create A-State organizers for their tremendous work to get this information available to the A-State community. Thank you very much.
Hello, my name is Maud Kutaiba Abu Ghazle. I am a PhD student in Environmental Science program. My project is to test the absorption of catechol and hydroquinone on the surface of TiO2 and iron oxide. I would like to take a moment to thank my faculty advisor, Dr. Hashim Ali, the chair of chemistry department, Dr. William Burns, and the environmental science program director, Dr. Tanya McKay, for their assistance in this project. Here is my Create A-State poster, and I will go through it later. As a result of increasing of human activities and energy-related CO2 emissions, climate is changing. And we can see that the temperature is increasing all over the world. The sea level rise, ocean warms, and there's a change in precipitation patterns and droughts. Also, there's already a scarcity in fresh water globally, where from all the water on the earth, there's almost 3% is fresh water, while 1% of this fresh water is surface water. The need for wastewater treatment is increasing to reduce the burden on fresh water. Catechol and hydroquinone are emerged from the usage of personal care products, which can be seen in household and industrial wastewater. In high concentration, these chemicals can cause severe human, animal, and environmental impacts. The first objective of my research is to investigate the absorption characteristics of organic pollutants, which is the catechol and hydroquinone, on the surface of titanium dioxide and iron oxide in aqueous solutions. The second objective is to test the effectiveness of the titanium dioxide and iron oxide as adsorbents for these chemicals. Fourier transform infrared spectrometer was used to accomplish this project. The tested pollutants was the catechol and the hydroquinone, which we can see that both of them, they have the same formula, C6H6O2, but the change of the location of the OH bond for each of them change the properties of these chemicals. These chemicals have been used in industry and pharmaceutical, especially in personal care products. It is found that these pollutants have harmful effects on humans, environment, and animals. Titanium dioxide and iron oxide was used as adsorbents in this experiment. Both of them have many applications in industry, and they attracted scientists to the research mainly because of their large surface area. FTIR, attenuated total reflection crystal spectroscopy, can provide data that gives insight on the structure of the compounds, as well as how strong the bond between the contaminants and the oxide themselves. The experiment took place when the hydroquinone or the catechol were exposed to the titanium dioxide or the iron oxide in several vials, as we can see here. And then the hydroquinone and the catechol was removed from the vials and placed on the attenuative total reflection crystal to be analyzed. The infrared spectra of catechol and hydroquinone was analyzed to see which peak was ideal for determination of adsorption kinetics on the surface of the oxides. The concentration of catechol and hydroquinone was increased, and the area under the curve was analyzed and plotted as we can see here. The slopes of the graphs were used to relate the integrated absorbance to the concentration of the catechol and hydroquinone. Through this project, we try to find the impact of pH on the adsorption capacity through finding the point of zero charge of the solution, positive or negative charge surface of TiO2 and Fe2O3, which determined by the solution pH and their point zero charge. The geometry of catechol and hydroquinone when adsorbed on TiO2 and Fe2O3 surface were found to present positive or negative effects on the adsorption capacity and its rate, depending on the pH point zero charge. The kinetics of adsorption of catechol and hydroquinone on the oxide surface was followed as a function of time. Adsorption kinetics reveals that with 99% certainty that the adsorption of catechol and hydroquinone on TiO2 and Fe2O3 follows a pseudo-second order model with equilibrium achieved within 24 hours. This means that the kinetic model is only affected by the initial concentration of catechol and hydroquinone. After validating the best model of all adsorption isotherms, the long-mer adsorption isotherm model shows the maximum adsorption in hydroquinone, while Freundlich isotherm was best for catechol on Fe2O3. Using iron oxide, catechol and hydroquinone follow Freundlich isotherm, while using titanium dioxide, the long-mer model was the best fit for catechol and hydroquinone. This is maybe due to the position of OH bond and the nature of the ring influence the binding mood on solid surface. TiO2 is seen as a good adsorbent for catechol and hydroquinone, indicating a strong chemical binding on the surface. Blending catechol and hydroquinone is ideal to be removed by TiO2 from wastewater. Here is a few references for my research project. If you need more references, it's upon request.
Hello, my name is Nicholas Craddock, and I am a graduate assistant under Dr. Jennifer Bolden, and my research project is the pasture use effect on water quality of subwatersheds of the Buffalo National River in Arkansas. Just a little bit of background, the Buffalo National River was designated as an extraordinary resource waterway by the Arkansas Department of Environmental Quality in 2008, and it earned this designation due to its primary and secondary contact activities and because it is economic resource to local municipalities. Of the subwatersheds of the study, 51 to 68% of the land surrounding them is forested, while 16 to 28% of the remaining surrounding land is used as pastures. It is a known fact that runoff from pasture land has both direct and indirect negative impacts on water quality parameters, such as pH, dissolved oxygen, nutrient loading, fecocoliforms, etc. Objectives of this study are to measure water quality parameters of each subwatershed and identify differences in water quality at the headwater and outlet site of each subwatershed, and in the end to determine if subwatersheds meet Ozark Highland State water quality standards. To do this, we took once weekly samples at the headwater and outlet site of 4 different subwatersheds, these sites being Bears Outlet Creek, Brush Creek, Calf Creek, and Tomahawk Creek. To get this data, we would use a bucket to collect water, and then in the field, we would measure parameters such as pH, temperature, conductivity, and dissolved oxygen. and flow. And then when we got back to the lab, we would analyze the data and the sample for turbidity and total suspended solids. Through the analysis of data collected, we found that for the parameters pH and conductivity, every sub-watershed has a significant difference between the upstream and the downstream sites, except for Tomahawk Creek, though for conductivity, the only site that had a significant increase between the upstream and downstream site was Calf Creek, with an increase in conductivity between headwater and outlet, while Bear Creek and Brush Creek both had a decrease in conductivity between the upstream and downstream sites. Analysis of turbidity showed that Bear Creek and Brush Creek were the only subwatersheds that had a significant difference between the upstream and downstream sites, even though the turbidity decreased on a downstream gradient for both of those sites, which we are currently trying to come up with a reason why that may have happened. But at the same time, Brush Creek was the only subwatershed with a significant difference in total suspended solids between the upstream and downstream sites, and as in the case of turbidity and conductivity, there was an actual decrease between the upstream and downstream site. This is an ongoing project, and we are still analyzing nutrient data and counting fecal coliform data, and will continue to do so for the duration of this project. So more data may become available and may change our overall outcomes, so we will see what happens there. But as of right now, I would like to thank my partners in this project, which are Dr. Jennifer Bolden and Jesse Brown, along with our funding for this project, which comes from the United States EPA and the Arkansas Natural Resource Commission, and as well as the National Park Service for their assistance with locating the sites and keeping the roads clean and accessible so that we can continue to get to our sites and collect data. So, thank you for listening.
Hello everyone, my name is Gaurav Gujral and I am a master's student in molecular bioscience, and I have been working with Dr. Fabricio Medina Bolivar under his supervision since past 7 months. And the topic of my poster presentation is biotechnological tools to study the role of phenylated stabinoids against oxidative stress in peanut. This is the overview of my poster presentation, which I am going to break down into pieces and explain. Before I go in depth into the presentation, I'd like to talk about a mature peanut plant. This peanut plant has huge economical importance due to its application in human health benefits and consumption need as well. The peanut plants are grown from a seed which gives rise to a mature peanut plant with flowers. These flowers then penetrates the soil and give rise to peanuts under the soil. And this is an interesting fact. During the growth of peanut, it faces a particular stress called drought in particular, which results in formation of different oxidative stress, which causes death of the plant. And this phenomena causes loss of around $520 million annually worldwide during production of peanut plant. However, in peanut plant, they have a response to stress such as drought by activating a defense mechanism which results in production of natural compounds to fight back against this stress. But the limitation is that there is limited availability of these natural compounds. However, in our lab, we have developed an artificial system called peanut hairy root, which can be used as a production platform for these natural products, which can fight back against oxidative stress such as drought. So let me explain how these hairy roots are produced in our lab. The leaf of peanut plant is infected with certain type of bacteria which results in formation of roots on the leaf of the plant, and these roots can be grown in liquid media in lab condition in a flask, and these peanut hairy roots that can then be stressed by using different chemicals which then produces the natural products in the media, and this is indicated by the color change in the media, and the natural products can then be extracted from the liquid media and can be tested for further use. So there are certain genes which are responsible for production of these natural compounds, and exploring the roles of these genes can help us identify the roles of these compounds against oxidative stress such as drought. The overall hypothesis of this research is that peanut hairy roots will increase the production of natural product called prenalated stilbenoids upon exposure to oxidative stress such as drought. To test this hypothesis, we will be carrying out two specific aims. The first one is to create transgenic peanut plant by downregulating the expression of genes responsible for production of these natural products. And the second one is to validate gene knockdown and investigate the antioxidant property, which is the defense mechanism used by these transgenic peanut hairy roots. So far in my research, I have developed a regeneration media protocol in which I have taken different parts of the plants and then grown them in media with different concentration of growth regulators. The media supplies them nutrient for their growth, whereas the growth regulator enhances shoot formation. And in my experiments, I have got shoot formation from two different type of growth regulators at specific concentration. Both the growth regulators resulted in formation of three shoots, and the growth regulators were benzylaminopurin and thidiazuron, which are both growth hormones. I have also established the hairy root culture for Tifroner cultivar with the procedure which I had described before by infecting leaf of peanut plant with certain type of bacteria. So far in my research, I have developed a regeneration media protocol for Tifroner cultivar and established hairy root line for Tifroner cultivar as well. And the further experiments that are remaining to be carried out in my research are to create transgenic peanut plant by knocking down the genes responsible for production of those natural products by using a biotechnological tool called RNAi, and then I will have to validate the gene knockdown of those genes, and then finally I will determine the antioxidant capacity of natural products synthesized from transgenic peanut hairy root. Thank you everyone for your time and stay safe.
Hello everyone, I'm Shankalpa Chakraborty, a first year graduate student in Molecular Biosciences graduate program. I'm working under the supervision of Dr. Vishwanathan Rajagopalan, and today I will be presenting our research on aberrant cardiac long non-coding RNA profile following altered thyroid hormone levels. Here is our poster. For better visibility and clarity, I will break it down into its major components. Cardiovascular disorders are number one killer not only in Arkansas and the entire United States, but also in the world overall. With 40% of the ongoing coronavirus COVID-19 pandemic patients dying with cardiovascular complications, our research becomes highly crucial and urgent in these unprecedented times. Abnormalities in thyroid hormones can adversely cause or contribute to cardiovascular diseases. Thyroid hormones are the master controller of the cardiovascular performance. There are two types of thyroid hormones, T3 and T4, both released from the thyroid gland. Dr. Raj Lab has published multiple papers showing beneficial effects of thyroid hormones in cardiovascular diseases. We are now on to discovering even some better therapeutics. Before going to the RNAs, the central dogma of biology states that DNA, which consists of innumerable genes, produces RNA, which in turn produces protein. But if we code all the proteins back to the DNA, it covers less than 5% of the total DNA size. So it becomes crucial to study the less explored non-coding regions. We call these RNAs non-coding RNAs. One such variety is called long non-coding RNAs, which are relatively new and emerging molecules to explore in cardiovascular biology, as evident in the picture. They comprise of at least 200 nucleotides, which in turn are the building blocks of DNA and RNA. Our hypothesis is that abnormal alterations in thyroid hormone levels will impair long non-coding RNA gene expression in the heart. The various techniques expanded here include gravimetry, ultrasound ECG scan of heart, as well as qPCR for long non-coding RNA expression and ELISA for thyroid hormone level, both of which are used to detect coronavirus COVID-19 patients. We also did exosome isolation and Western blotting, as well as statistical analysis. We first established hypothyroidism or low hormone levels and hyperthyroidism or high hormone levels in mice using excessive amounts of compounds called PTU and T4 respectively. This slide demonstrates that with hypothyroidism, the heart and its components reduced in size, and with hyperthyroidism, they increased in size. As is tested for all heart patients, including those with coronavirus COVID-19, hypothyroidism reduced heart function and decreased heart rates. Hyperthyroidism, on the contrary, showed opposite functionalities. Importantly, RNA purified from the heart tissues of these animals show that long non-coding RNAs are significantly impaired in both hypo and hyperthyroidism, especially in a differential and opposing manner. Since detection of long non-coding RNAs in blood can serve as an excellent diagnostic tool, we isolated exosomal vesicles, which are major carriers of the long non-coding RNAs in blood serum. As a proof of principle, I was successfully able to isolate and then confirm the enrichment of exosomes through Western blotting technique using CD63 antibody. In summary, this is the first work of its kind detecting heart-specific long non-coding RNAs following altered thyroid hormone levels. We strongly believe that these findings, along with the future expansion of exosomal studies, will enable us to discover novel diagnostic and therapeutic strategies for not only coronavirus COVID-19 patients, but also all patients with cardiovascular disorders. Following are the references that we have used for this presentation. We would like to acknowledge and thank Arkansas State University, Arkansas Biosciences Institute, and NYIT College of Osteopathic Medicine for their support. Last but not the least, thank you so much for your attention.
Hello everyone, I hope everybody is doing well. My name is Sharon Podell and I'm doing my master's in molecular biosciences here in Arkansas State University. I'm currently working under Dr. Esela Winzaratne. He is my mentor and research advisor. Today I'm here for the poster presentation of one of my current research work. And the topic for my research is reduced representation by sulfide sequencing to detect condition-specific DNA methylation in soybean. Our lab, we specifically focus on soybean because it is one of the most important crop in United States for its nutritional value, export to different other countries, and diverse uses from the food industry to the textile industry. However, due to various biotic and abiotic stresses like pathogen infection, temperature, radiation, and water, we lose tremendous amount of yield annually. During these stresses, plant goes through different genomic modification and DNA methylation is one of them. DNA methylation can affect plant production, survival, and development as well. So we can define it as an as an epigenetic modification which occurs when an enzyme called methyl transferases adds methyl group in the 5th position carbon of cytosine. Cytosine is one of the building block of DNA, and in figure number 1 you can see how DNA methylation happens. DNA methylation can directly affect the gene expression by switching off the gene by inhibiting RNA polymerase, which is an important enzyme for DNA transcription, to promoter region of that gene. And in figure number 2 and 3, we can see how gene silencing happens due to the inhibition of transcription due to cytosine methylation in promoter region. So study of methylone profile of a plant gives us an insight to elevate stress tolerance and increase crop productivity even during adverse conditions. In our research, one of our objectives is to compare whole genome bisulfite sequencing method, which is our conventional method for methylome profiling, with reduced representation bisulfite sequencing method, which is a novel method for methylome profiling. Even though both of the methods uses bisulfite converted DNA fragments, whole genome bisulfite sequencing uses random fragmentation method and sequences whole genome, but RRBS uses specific endonuclease restriction enzymes for selective fragmentation of DNA, and only those fragments are used for sequencing. So these differences make RRBS more cost effective and also more specific compared to whole genome bisulfite sequencing. Our second objective is to optimize RRBS library preparation method. You could see in figure number 5 under materials and method, both methods are similar. However, for whole genome bisulfite sequencing method, random fragmentation can be done by using sonication, and for RRBS method, specific endonuclease restriction enzyme such as MSE1 it can be used to cleave the genomic DNA in specific sites. So during library preparation, adapter ligated DNA fragmentation fragments were selected accordingly to their sizes, and then bisulfite conversion was done where non-methylated cytosine were converted into uracils. So moving towards the results included in this poster are preliminary data which only includes one RBL sample. In figure number 6, you could see you could visualize the smear of DNA fragment size between 200 base pair to 450 base pair length in the gel after gel electrophoresis, which tells us that the MSE1 worked very well as a restriction enzyme for soybean genome. And after receiving the sequence result from the sequencing facilities, quality assessment of data was done using FASTA QC and Fred box plot method, where in figure number 8, you could see that the Fred score for our sequence is more than 20 up to 40, which indicates that the quality of the sequence is very good enough for the downstream steps like cut adapt to get rid of adapters, Bismarck for methylome profiling, and methylkit for the final the data processing. So let's move towards other results we obtained after aligning, or we can say comparing our sequence with database online. In figure number 9, we can see that the high methylation was observed in cytosine guanine dinucleotides, followed by CHG, CHH, and CHN context, H being nucleotides other than cytosine. And in figure number 10, it shows that 75% of genes from our sequence method were matched with the database. And also in table number 1, we could see that the 59.6% of reads were aligned with the database. And with all these results, we can conclude that 75% gene mapped with the database. In overall, MSE1 was a good choice as a restriction enzyme, and in future we need wider size range of DNA fragment to include more genes. And in coming future, our goal is to compare these two sequencing methods and use different other restriction enzymes to make RRBS protocol more specific. And finally, we want to thank A-State internal seed grant for the funding, and Arkansas State University and Arkansas Bioscience Institute for the support and facilities. Thank you very much for listening to me, and have a nice day.
Hello, my name is Jennifer Terry. I am a graduate student mentored by Dr. Lori Newman-Lee, and today I will present endocrine and immune patterns in freshwater turtles at Barradish Farms, Arkansas during the active season. All organisms must allocate finite amounts of energy to various processes through trade-offs. These trade-off dynamics are context-dependent on the individual and the environmental level. Glucocorticoids are expected mediators of these trade-offs. As an example, the innate immune system is then influenced by these endocrine patterns. Unlike birds and mammals, turtles and other reptiles must get their heat from an external source. Turtles have long lifespans and many reproductive opportunities. There's a disparity between males and females in terms of reproductive obligations, and turtles rely primarily on the innate immune system. For my project, I compared endocrine and immune responses to an acute stressor across demographic groups throughout the activity season. We captured red-eared sliders for 7 to 10 days each month between May and October using baited traps. We collected blood samples, morphometrics, sonograms, and then used this plasma collected from the blood to perform bacterial killing assays and radioimmunoassays to assess innate immune function and corticosteroid concentrations. I then conducted Kruskal-Wallis and post-hoc Dunn's tests using Program R. Bacterial killing ability, or a measure of the innate immune function, varied in females across months. In particular, there were lower levels of killing ability between May and July compared with August and October. There was no significant difference between bacterial killing ability from samples collected at the 30 and 60-minute time points. When grouped by sex, bacterial killing ability was lower in juveniles than adults, as shown in Part A of Figure 5. Circulating corticosteroid concentrations, however, were higher in males than they were in females and juveniles, as indicated in Part B of Figure 5. It is also important to note that females had relatively no change in circulating corticosteroid concentrations throughout the acute stressor. In conclusion, the response to an acute stressor varies by sex and time. Males may have access to more energy to mount an appropriate response to stressors than females and juveniles. Juveniles are likely still developing their immune system and also are diverting more energy towards growth. Females are likely mobilizing a lot of energy toward clutch development and nesting in May through June, are then deprived of energy by the end of the nesting season in July, and then regain energy throughout the late summer into fall through foraging. This may explain why we saw higher levels of bacterial killing ability between August and October compared with May through July. In addition, the high levels of baseline bacterial killing observed August through October may be sufficient. For the same reasons, energy is likely not prioritized toward the court response, explaining why we saw no change throughout the acute stressor. Across sexes, other traits within the individual may also influence responses. For my future work, I hope to complete my radioimmunoassays, conduct reproductive assessments, include morphological data in my statistical analyses, and add additional field seasons to my data set. I would like to thank my undergraduate field technicians, my mentor, Dr. Lori Newman-Lee, and my various funding sources, including Arkansas State University. I'd also like to thank you for listening to my presentation today.
Hello, I'm Karen Bounds from Brooklyn High School. I'm happy to present at Created a State as a YES Scholar conducted by the Arkansas Biosciences Institute at Arkansas State University. I work under Mr. Sankalpa Chakraborty in the laboratory of Dr. Viswanathan Rajagopalan. Today I'm going to discuss our exploration of epigenetics in the regulation of cardiovascular diseases. I would like to start off by giving insight on epigenetics. Epigenetics is the study of heritable changes that do not alter genetic sequences of deoxyribonucleic acid, also referred to as DNA. Several epigenetic processes exist, but this experiment focuses on histone acetylation due to its possible link to CVD. To better understand this, we will explore what histones and histone acetylations are. Histones are proteins that act as a spool which DNA strands wrap around due to an attraction between the two as they have opposite electrical charges. Histone acetylation functions to alter this charge with the addition of an acetyl group to the amino acid lysine at one end of the histone molecule, causing the DNA to unravel from the histone. This exposes more docking sites for regulatory proteins, causing genes to undergo or halt the transcription of ribonucleic acid, also known as RNA. This experiment attempts to discover a relation between histone acetylation and CVD. CVDs are the #1 killers of humans, not only in Arkansas but worldwide, and recent studies have shown a link between epigenetics and CVD. Exploring histone acetylation's relation to CVD could lead to better treatment strategies. To determine a link between the two, heart tissue samples are taken from 3 separate groups of rats are examined. These 3 groups include a control group, a thyroid hormone abnormality, CVD group, and a CVD group treated with thyroid hormones. Thyroid hormones are important to this experiment because thyroid hormonal abnormalities can cause heart disorders. These tissues were examined using electrophoresis and Western blotting methods, which I will explain shortly, to determine levels of histone acetylation in each group. The first process is subcellular fractionation, which is the purification of homogenates or liquid samples containing all cellular organelles, using a centrifuge, which is a machine that separates homogenates using centrifugal force revolutions over a given period of time. We use this to target nuclear proteins in our heart tissue samples. The next step begins SDS-PAGE, which abbreviates sodium dodecyl sulfate polyacrylamide gel electrophoresis. Using the detergent sodium dodecyl sulfate, we straighten the folded protein so separation is by size and molecular mass only. Next, we conducted polyacrylamide gel electrophoresis, which uses the positive charge to pull negatively charged proteins through the gel, allowing proteins to be measured. The semi-dry Western blot process is next. This process transfers proteins from the gel to a PVDF or polyvinylidene fluoride membrane. We will do this because the gel is not a suitable platform for antibodies to bind with proteins, but the PVDF membrane is. The gel and PVDF membrane will be placed adjoining one another between 2 filter papers which have been soaked in a transfer buffer and placed in a semi-dry transfer system which uses a positive electrical charge to attract proteins from the gel into the PVDF membrane. Next, we will wash the membrane in a blocking buffer called bovine serum albumin, or BSA. This washing fills all empty binding surfaces, preventing the nonspecific binding of antibodies. This means these antibodies only bind to the targeted protein, providing accuracy. We will then wash the PVDF membrane in primary and secondary antibodies, which are commercially available rabbit antibodies raised against the target H3 acetylation proteins in rats. The PVDF membrane is washed in primary antibodies which bind to our target proteins, and then secondary antibodies which bind to each primary antibody and are detectable when developed in an imaging system because they were incubated in a dye and contain an enzyme or fluorophore, which is a light-emitting chemical. After this, the PVDF membrane will be washed in a developing reagent which will react with the dye and secondary antibodies to emit light, which is detectable when the PVDF membrane is developed in an imaging system, allowing us to see the concentration of H3 acetylations. We expect there to be a significant difference in the amount of H3 acetylation status among control and experimental groups. As described in this poster, exploring the status of H3 acetylation in cardiovascular disease can shed light on better understanding how to treat the disease. I thank the opportunity to present in this prestigious symposium, and I aspire to discover major new findings towards better diagnosis and treatment of these lethal disorders. So far, I have been able to perform serial dilution of test albumin proteins, run SDS-PAGE, stain with Kumasi, and destain it subsequently. As soon as the COVID-19 pandemic ends, I look forward to continuing the rest of this process.
Hello,
I am Susan Hanrahan, Dean of the College of Nursing and Health Professions. I am very excited that our college has put forward over 75 poster and oral presentations for this year's Create at State. I know we usually have well over 100, but I'm pleased that our students and their mentors have agreed to participate in this actually very unique and exciting virtual forum. In our college, I think you'll see an eclectic array of research, which can involve either highly specialized equipment, survey research, or other methods of research. Most of the research that you'll see listed actually has direct application, so that's the cool thing about it. We can do quality assurance projects, we can do quality improvement projects, and all of these things, actually we can use federal guidelines or national guidelines and do comparative analysis to things that are going on in the clinics so that we can see what may actually impact clinical practice. Patient access, patient outcomes, quality of care, etc. So it really is a win-win for everybody involved when we do these applied research scenarios. Some of our graduates won't finish their research until the spring or actually this summer, and I wish that they could participate because it would even demonstrate more diversity within the college. But this year I want to thank all of our students who took the challenge to go virtual and their mentors, and so we do still have a quality showing of our research talent in the college, and I'm very proud of all of our students and our faculty who are participating in this unique forum. So, from the College of Nursing and Health Professions, everybody enjoy.
Hello, my name is Joseph Austin, and my presentation is titled Teachers' Perception on Screen Time in 5th and 6th Grade Classrooms. First, I would like to acknowledge Dr. Krista Susie Snowgrove, my advisor, and also Dr. Molly Manning and Miss Melanie Mata. Over the past few years, technology has been integrated in nearly all aspects of life, including our school systems. The American Association of Pediatrics has put out recommendations to have consistent limits on screen time in children ages 6 and older. Now, screen time is defined as a term used for activities done in front of a screen, such as watching TV, working on a computer, or playing a video game. Research has shown that teachers have varying views on the use of technology in school. They think that it can either be a distraction or that it can cause students to be more well-rounded. My research question is, how do teachers believe screen time at home and in schools affects school performance in 5th and 6th grade classrooms? I have two hypotheses with this research question. The first is that teachers will be split on the topic of screen time in schools based on age and what subject they teach. The second being, teachers will agree that screen time at home decreases school performance. Current research indicates that screen time is linked to many physical health problems. Due to the sedentary behavior associated with screen time, research has linked it to an increase in prevalence of obesity, decreased muscle mass, sleep problems, and musculoskeletal pain. It has also been linked to mental health issues. There is a positive correlation between depressive symptoms, stress, anxiety, and increased screen time. The only exception to this is a negative correlation between binge watching and severe depressive symptoms. Research shows that recommendations on screen time are not widely known. Studies aimed towards parents showed that very few know screen time regulations or enforce those regulations at home. Teachers generally share one of three views on screen time in schools. They either think that it is a distraction to students during school, that it can help students learn in an individualized manner, or that it impacts students' long-term development. Participants included 13 teachers actively teaching a 5th and 6th grade classroom over the age of 18. Participants were recruited from publicly available email addresses according to school websites. Schools utilized were located in Northeast and Central Arkansas. A 14-question survey was sent through SurveyMonkey, analyzing demographics, technology utilized in classrooms, and opinions on trends in school performance based on screen time. The survey was emailed to participants in January of 2020, with a reminder sent 2 weeks later. The survey remained open for 2 months before results were analyzed. "60 teachers were emailed the survey. Of those, 13 completed it. The average age of the respondents was 35 to 44 years old, which composed 30.8% of the group. Additionally, 30.8% of the group had been teaching for 20 or more years. An analysis of the results showed that teachers have varying beliefs on how screen time impacts school performance. 5 of the respondents indicated that if screen time in schools decreased, the performance would increase. 4 teachers believed that school performance would decrease if screen time in schools was decreased. Additionally, 4 teachers believe that screen time has no significant impact on school performance. An analysis of this data showed no correlation between age or number of years teaching and opinion on screen time in schools. 80% of math teachers who responded indicated that screen time in schools could be beneficial to students. No other subjects taught showed a consistent opinion. In contrast to differing opinions on screen time in schools, teachers showed more unified opinions on how screen time at home impacts school performance. 85% of respondents indicated that if screen time at home was decreased, school performance would be increased. The remaining 2 teachers indicated that school performance would not be impacted by a decrease of screen time at home. The hypothesis that teachers will be split on the topic of screen time in schools based on age and what subject they taught was proven wrong. The study showed no indication that age was a factor in opinion on technology in schools, and the only subject that showed unity in opinion was math. However, the hypothesis that teachers will agree that screen time at home decreases school performance was proven correct. The researcher recommends that the Arkansas State Board of Education creates clear guidelines on screen time in schools to comply with the American Association of Pediatrics and to provide teachers with a clear goal. More research is needed on how school performance is truly impacted by screen time trends. Here are the references used in this project, and thank you for your time.
Yes, this is a policy analysis of the 2017 ASA NPO fasting guidelines among GI surgical patients. Tony Banks is the main researcher. Faculty member is Dr. Christopher Jordan. Yes, the inspiration for this project was my mother, who was an 85-year-old lady, who was diagnosed with cancer and needed to have a peg tube placed. She had already been NPO, nothing in my mouth for gallbladder surgery, and now would not be allowed anything further until after the peg tube placed the next day. For an elderly lady, this would be over 24 hours of NPO status. The question is, why is there such a long time to become NPO prior to surgery? The primary purpose of the fasting guideline is to improve the quality and efficacy of anesthesia being provided to the patient. Its principal mission is to reduce or eliminate perioperative pulmonary aspiration during the pre- or post-operative period. It's based on 2 main principles discovered in research. The ingested material must be clear without particulates, and it must not be within the 2 hours of the procedure. Research question is, does educating the medical community on the updated fasting guideline decrease patient delays and cancellation of surgery and improve patient comfort? The review of the literature have proven that it does. In all, 499 patients were included in the study. 289 were females, 210 were males. The average age of the patient was classified in 4 classes: young, middle age, elderly, geriatric. The average age is 60 years old. The majority of patients, 40.5%, were elderly patients, followed closely by the geriatric, 39.7%, followed by the middle age, and lastly by the young patients. The research study was conducted to explore the relationship between NPO violations, practice guideline violations, age, ethnicity, and gender in centers A, B, and C. We did not observe any relationship between NPO violation and age, ethnicity, and gender in centers A, B, and C. We also did not find any connections between the practice guideline violation and ethnicity and gender at centers A, B, and C. However, we found that there was an association between the practice guideline violation and age of Center B. In other words, the practice guideline based on their age, that is, age played a role in the violation of practice guideline at Center B. To confirm this, we computed some correlation between the raw scores of age and time. We found there was a weak negative correlation between the age of the patient and the amount of practice guideline time violation at Center A, B, and C. For the NPO time violation and age, there's a very weak negative correlation in centers A, B, and C. The average NPO violation times in centers A, B, and C was 4 hours and 8 minutes, 4 hours and 25 minutes, and 4 hours and 16 minutes respectively, all greater than 2 hours. So on average, there was no NPO violation in center A, B, and C. The average time for practice guideline violations at centers A, B, and C was 5 hours and 38 minutes, 5 hours and 54 minutes, and 5 hours and 31 minutes respectively. The financial implication of the study is directly related to the number of cases that are delayed or canceled. The facility had 20 cases canceled last year due to NPO violation. They were canceled due to ingestion of substance requiring 60 more hours of additional NPO time. Broken down into quarters, it was 7 cases, 2 cases, 5 cases, and 6 cases respectively. Yes, the loss of a single case due to NPO violation cost the center between $1,726 to $2,254, depending if it's a colonoscopy or EGD. If it's a self-pay patient, $1,075 to $1,482, depending on type of case. Total case for the loss of 20 patients, $34,520 to $45,080 if the patient had private insurance versus $21,500 to $29,640 if it's self-pay, or $238.58 per hour that staff went over. The recommendations from this research of the study is to have all patients adhere to the 2017 ASA practice guideline 2-hour NPO policy. This would still allow the 3-hour NPO window set forth by the facility anesthesia profession as demonstrated by the research study. By naming the policy change, you can theoretically decrease, on average, the NPO time from 1 hour at all 3 centers from 4 hours to 3 hours, thus achieving the 3-hour window sought after by the anesthesia staff. There would also be a theoretical decrease in the practice guideline recommendation at all 3 centers from 5 hours to 4 hours, once again achieving the goal of 3 hours set forth by your anesthesia team before the surgical procedure.
Hi, my name is Amber Clements Four. I'm a DNP student at Arkansas State University. The title of my project is Knowledge, Attitude, and Practice of Nicotine Use Among Youth. So my research question is, will implementing a parental teacher education class regarding nicotine use among youth in rural areas improve awareness and decrease nicotine use after completing a community awareness presentation of the dangers of nicotine among children? My DMP translation project had two aims. The first, to establish the number of teenagers in the 9th through 12th grades in rural school districts who have ever used nicotine products. And number two, to establish knowledge of nicotine use among youth from the perspective of parents and educators. My objective was to evaluate awareness of nicotine use, delivery of electronic cigarettes and Juuls among youth, and to develop a plan of cessation. The design of my project used two existing tools, one developed by the CDC and one developed by Dr. Jessica Pepper from the University of North Carolina. These were used to evaluate knowledge, skills, and attitudes of use of electronic cigarettes. There was a pre and post survey that was delivered in conjunction with an educational seminar given to educators and parents. A survey tool, which was used by the CDC, was delivered via SurveyMonkey to high school students to evaluate the knowledge and use of electronic cigarettes. The setting of my project was at Cave City High School, which is a rural area in Arkansas. It involved two populations: high school students, grades 9 through 12th, and high school staff and educators and parents of high school students, 9 through 12th. Excluded populations were those students that were not in the high school and those that were not parents or educators affiliated with high school students. Informed consent was included on both surveys. So unfortunately, the results of my SurveyMonkey on high school students were not surprising. 93% admitted to have heard of Juuls or electronic cigarettes. 54% said they had used these devices. 11% said they used daily. 15% said socially. 11% of these children said they had received their devices from parents or another adult. However, 91% said they do not think that vaping is healthy, and 73% that use said they would be willing to quit. And the gender was 52% male and 48% female, and it was grades 9 through 12. On my adults, educators, and parents, 56% were female, 44% were male, 100% non-Hispanic. 10% said they were smokers, 90% said they were non-smokers. 0% were current vape users. 3% they said they had used in the past. 100% said they learned about electronic cigarettes from the media, and 8% said they know nothing at all, with 13% saying they knew quite a lot. However, 85% said they would be interested in learning more about electronic cigarettes. On the post survey, the knowledge did change quite a bit. So when it says, would you be interested in learning more about electronic cigarettes? 90% said yes. How much did you know about electronic cigarettes? It went from nothing at all at 3% to quite a lot at 46%. The belief and attitude portion of the post surveys also changed quite a bit. Electronic cigarettes are safer to use than regular cigarettes. 0% strongly agreed. Electronic cigarettes are a helpful aid in smoking cessation. 0% also strongly agreed. 47% think that electronic cigarettes may be a conventional gateway to cigarette smoking. 82% said that they do think that it's a public health concern. 79% think that electronic cigarettes should be regulated in work and public places. And 85% stated they were extremely likely to counsel a family member on electronic cigarettes. So lessons learned, need for future scholarship. There needs to be vaping cessation aids for youth To date, there are no pharmacological treatments available. There's a need for age-appropriate cessation tools. Early screening is also important. We should incorporate this in all sick visits and well-child visits, begin with elementary and middle school ages, and we need to collaborate and improve knowledge, belief, and attitudes with our community leaders, school officials, and parents.
Hello, my name is Kirsten Combs, and I'm a graduate student in the Communication Disorders program. This is my thesis titled Speech-Language Pathologist Knowledge and Implementation of Thyroid Hormone Diseases and Allergies on Voice. My mentor was Dr. Amy Schollenberger. About 5 years ago, I was diagnosed with hypothyroidism, thyroid nodules, and I began weekly shots for my allergies. And since then, I have noticed many changes in my voice. This led me to my research question of what do speech-language pathologists know about the impact of thyroid hormone diseases and allergies on voice disorders, and is this knowledge demonstrated in their case history questions for voice clients? My thesis has two hypotheses. First, that there will be a positive correlation between SLPs who know how allergies affect voice and SLPs who include allergy questions in their case histories. My second one is that there will be a negative correlation between SLPs who know how thyroid hormone diseases affect voice and SLPs who include thyroid disease questions in their case histories. A voice disorder is the difference between an individual's voice and what is normal for an individual of the same age, gender, culture, and geographic location. The term allergy is a chronic condition in which the bronchial tubes narrow and cause inflammation when exposed to a substance. In a study of 80 participants, almost half of them had a voice disorder, and they found that allergies were associated with diminished hearing and misarticulations. That I could find, there are no previous studies concerning SLP inclusion of allergies on case histories or their knowledge of allergies. A thyroid disease occurs when there are abnormalities in the thyroid gland's hormones that affects the gland's structure and/or function. These individuals present roughness, glottal fry, strained vocal quality, breathiness, and pitch disturbances. The participants in the study were SLPs in Arkansas that were ASHA certified. For the materials, a 24-question survey was generated using Qualtrics. The questions addressed demographics, thyroid hormone diseases and voice disorders, allergies and voice disorders, and questions regarding SLPs, case history, question routines for thyroid diseases and allergies. If the participant reported that they were not ASHA certified on the first question, they were then directed to the end of the survey. Also, an email list of SLPs were generated from a VESPA. After the survey was made, an email was sent to every SLP from the ABESPA email list, inviting them to participate in the attached survey. After 1 month, an automatic reminder was sent to those who had not completed the survey, and the survey stayed open for 2 months total, and then the data was analyzed. 387 certified SLPs accepted the survey, but only 284 actually completed it, meaning the total number of participants was 284. As you can see, most of the participants have been an SLP for more than 20 years, and most of them worked in a school setting or pediatric facility. Most of the participants knew which voice symptoms were caused by allergies, hypothyroidism, and hyperthyroidism, and most of the participants asked their clients who had allergies. However, very few asked them if they had a thyroid hormone disease, only 20. Of the 20, only half asked which disease the client actually had. Studies have shown that each thyroid disease causes different voice symptoms. The hypotheses of this thesis were supported by the data, and there is a need for more information regarding thyroid hormone diseases and allergies when treating voice clients. The correct diagnosis determines the effectiveness of treatment for our clients. A part of any health profession is improving our clients' quality of life, and studies have shown that these specific clients have a lower self-assessment of their voice, impacting their quality of life. So what are we doing for them? Here are just a few ideas to expand on this study in the future. First, a separate survey could be sent to endocrinologists and allergy specialists regarding voice symptoms and disorders to determine if these specialists refer their patients to SLPs for possible voice disorders. Also, a future study would be beneficial if SLPs were trained regarding how thyroid hormone diseases and allergies impact a person's voice, and at the end of the training, resurvey them to determine if their practices will change regarding case history questions and treatment. Endocrinologists and allergy specialists could also be involved to facilitate interdisciplinary practice. A third option would be a case study to be completed of SLPs co-treating with the endocrinologist or allergy specialist with the voice client. Here are the references that were used for this presentation, and thank you very much for your time.
Good morning. My name is Zelda Epperson, and I am currently a Doctor of Nursing Practice student at Arkansas State University. Dr. Todd Hammond is my program chair and mentor for this project, and the title of my presentation is A Comparative and Cost-Benefit Analysis, Vancomycin versus ANSEF Use in Total Knee Arthroplasties. A little background information for this presentation begins with a definition of total knee arthroplasty. This is a procedure to replace the joint in the knee and is the definitive treatment for osteoarthritis of the knee. Although our infection rates have decreased in the last decade, total joint infections are a complication from this procedure and can be a massive expenditure on healthcare. Costs can be anywhere from $19,000 to $68,000. This project is based on determining the correct preoperative antibiotic to prevent these infections and potentially save thousands of dollars for healthcare and patients. The methodology consisted of a retrospective review of 1,661 total knee arthroplasties from 2 rural hospitals within the Mississippi River Delta. A random sample number generator was used to generate a random list of integers as a comprehensive version. The sample size calculator was utilized to obtain a sample of 313 based on a 95% confidence level with a margin error of 5%. A Pearson's correlation was used to determine a relationship between the antibiotics and the comorbidities, obesity, smoking, diabetes, recent hospitalization, and previous methicillin-resistant Staphylococcus aureus infections. A two-tailed paired t-test was also used to compare the usages of vancomycin and ANSEF with readmissions related to the TKA infections. A Shapiro-Wilks test was used to test for a normality in the distribution, and the Wilcoxon-signed rank test was used to compare the comorbidities related to the readmissions related to TKA infections. The results showed no significant findings with the Pearson's correlations related to the comorbidities tested against each antibiotic, vancomycin, and ANSEF. A normal distribution was not found with the Shapiro-Wilks test. Therefore, a Wilcoxon-signed rank test was performed in order to reject the null hypothesis that there was no relationship found between the readmission related to the TKA infection and the comorbidities tested. The null hypothesis was not rejected for the recent hospitalization and the readmission related to the TKA infections. Cost savings associated with this study was related to ANSEF being the policy-driven antibiotic to be given preoperatively. All of the documented infections in this study received ANSEF preoperatively instead of vancomycin as the initial antibiotic. This study showed similarities with other studies in that there were no correlations between the readmissions related to TKA infections and comorbidities. There were 5 documented deep tissue infections over the course of the 5 years of total knee arthroplasties. Each infection cost approximately $47,000 when you factor in the readmission, the length of stay at the hospital, the cost of care, cost of antibiotics, laboratory testing, additional surgical procedures for possible incisional and drainage of the infected knee, removal of hardware, replacement of hardware, and any additional care. ANSEF cost the hospitals approximately $3.31 per gram, and vancomycin cost approximately $15.01 per gram, an additional $3,661 over the course of 5 years for the addition of vancomycin would potentially save the hospital $235,290 over the course of 5 years. That is a substantial savings. This chart shows a breakdown of the antibiotic cost and the breakdown of the infection cost per year. The limitations of this study was the cohort was based on only deep tissue infections, as this is all the infection control for the small hospitals track, and not the superficial infections or the cellulitis that may have also been reported to other hospitals. Another limitation is that the hospital is not able to track possible infections from patients that may have reported to a different facility. This study was also not generalizable due to the small sample size. Additional studies would be necessary with a much larger sample size to obtain accurate results. The implications from this study include updating our current guidelines with current information on the correct antibiotic dosage and administration as microbes change. Cost savings for the entire healthcare system is also an implication, as the cost of healthcare impacts everyone on some level, from the provider all the way down to the patient. These are the references that I used to complete the study. And thank you very much for your time. Any questions?
Hello, I'm Chris Ivy, and I would like to thank you for listening to my presentation of the project Opioid Reduction or Elimination with the Utilization of Transversus Abdominis Plane Blocks in Gynecological Surgeries. I would also like to make a special thank you to my faculty advisor, Dr. Sarah Davidson. I would like to start by giving a little background as to why I chose this topic. As a CRNA, we have a fundamental role in helping prevent pain, or at least decreasing it when possible. This has become an increasing challenge with the opioid epidemic our world now faces. Opioid abuse has become more concerning for providers in recent years. Providers have had to rethink and evaluate their opioid use for their patients. In 2017, there were more than 72,000 overdose deaths in the United States. I feel an often overlooked or underutilized area of anesthesia is that of regional anesthesia. With abdominal surgeries, one particular block has come to the forefront due to its ease of learning and efficacy. That is the transversus abdominis plane block, or TAP block for short. The research problem is, does the addition of the transverse abdominis block to gynecological surgical patients anesthetic plan decrease or eliminate the opioid need postoperatively during the first 24 hours? This is also my PICO question. The purpose of this retrospective study was to evaluate the efficacy of the TAP block in women undergoing gynecological surgery and the decreased or eliminated opioid need. The primary aim of this project is to demonstrate via pain scales how well the regional block, the TAP block, decrease the need for opioid in these patients. It should easily be measured using standardized pain scales at various times during the patient's stay. Primary objective is to eliminate or significantly reduce the need for opioids during the first 24 hours. This project is a non-experimental retrospective comparative analysis to determine or evaluate the efficacy of the transverse abdominis plane block and elimination or decrease usage of opioids within the first 24 hours following gynecologic surgery. Gynecological surgery patient records in 2018 and 2019 were reviewed to verify the utilization of the TAP block or not, also pain scores in the recovery room, and during the first 24 hours post-surgery were included. Demographic variables such as age, race were examined to describe this population. As previously mentioned, the methodology for this project is a retrospective comparative analysis. It is a quantitative analysis. We also use chi-squared for independence testing, and we use some descriptive statistics. For the chi-squared test of independence, a significance level of 95% or a p-value less than or equal to 0.05 was used to accept or reject the null hypothesis. The results of the chi-square test were significant, suggesting that the TAP block provided and opioid need in PACU are related to each other. Descriptive statistics and chi-squared for independence was utilized for this project. In all areas, the chi-square test does suggest that TAP blocks provided some degree of pain relief. By incorporating regional anesthesia into the plan of care of our patients, we are able to not only decrease opioid consumption, but in some cases eliminate the need altogether. This project, although small in number, does show promise that the hypothesis of the TAP block administration does, in fact, reduce opioid need for gynecologic patients during their first 24 hours postoperatively. The results of this project did show promise of being a good adjunct to the gynecological surgery patient's plan of care. There was correlation of having the TAP block and reduced opioid need in PACU and during the first 24 hours. This can potentially lead to decreased hospital stays, increased compliance, and increased overall satisfaction of the patient. Further reviews or studies would need to be made to establish correlations to the TAP block in these potentials. Value findings are important not only to the patient, but to that of the providers and the healthcare institutions themselves. Outcome findings are paramount that we review existing plans of care and look to see if any modifications are needed. Sustainability should be easily attained until better adjuncts are created. The entire premise of decreased opioid use, increased compliance, and satisfaction of the patient makes the project a win-win for all involved.
Hello, my name is Justin McAdoo. I'm a student in the Doctor of Nursing Practice program at Arkansas State University. This presentation focuses on a policy analysis of a substance use disorder policy for certified registered nurse anesthetist. My faculty mentor is Dr. Christy Black. Substance use disorders are the most common occupational hazard for CRNAs. An estimated 15% of practicing CRNAs will have a substance use disorder at some point during their career. Three major occupational hazards for CRNAs that impact the development of substance use disorders are drug accessibility, attitude, and occupational stress. Studies have shown that employers can avoid unnecessary and non-productive costs if evidence-based policies are in place that include offering their employees appropriate treatment and assistance with their substance use disorder. Anesthesia workplace policies and procedures are beneficial because they serve to identify the institutional goals and objectives, describe the strategy for achieving the goals, serve as a basis for future decisions and actions, control performance, and increase consistency of action. Few anesthesia groups have knowledge about the avoidable costs that an evidence-based substance use disorder policy can produce. It is vital for employers such as anesthesia groups to understand the impact that substance use disorders have on their organization and develop strategies that address the risk and costs associated with CRNAs with substance use disorders. This scholarly project seeks to answer if the substance use disorder policy for certified registered nurse anesthetists employed by lifelink anesthesia follow the current best practice guidelines. The purpose of this scholarly project is to analyze lifelink anesthesia's substance use disorder policy for CRNAs, compare it to the current best practice guidelines, and if applicable, present recommendations for policy revisions to key stakeholders. This policy analysis will seek to discover and address any evidence-based practice gaps associated with lifelink anesthesia's substance use disorder policy. The conceptual framework selected for this scholarly project is Eugene Bardak's eightfold path for policy analysis. Peacock approach was utilized to develop this scholarly project. Policy analysis was carried out. A review of the literature initiated the process by collecting all the currently available evidence-based research related to the current best practice guidelines. The current condition of lifelink anesthesia's substance use disorder policy was examined to determine its benefits and its risks. Lifelink's anesthesia's substance use disorder policy for CRNAs was compared to the best practice guidelines for a substance use disorder policy in order to discover any gaps between the current substance use disorder policy and evidence-based practice. Also, lifelink anesthesia's substance use disorder policy was evaluated for any possible revisions in order to align with the current best practice guidelines. Phase 1 involved performing a SWOT analysis of lifelink anesthesia's substance use disorder policy. The data collected during Phase 1 was utilized in Phase 2, which included a gap analysis between lifelink anesthesia's substance use disorder policy and the current best practice guidelines. Recommendations for revisions were developed, including evidence-based education related to substance use disorders, prevention strategies, treatment options, and support for CRNAs and their families. Educational recommendations include adopting evidence-based strategies for promoting the healthy well-being of CRNAs, adding appropriate language outlining the general and anesthesia workplace specific risk factors, and adding a list of signs and behaviors associated with impairment and drug diversion, adding to the substance use disorder policy a list of harmful consequences of drug diversion and substance use disorders in the anesthesia workplace environment. Also included are considerations for drug diversion prevention strategies. Additional recommendations focused on specific procedures such as random drug testing, workplace reporting, interventions within the workplace environment, treatment options, and reporting to the appropriate state board, reentry plans, contracts for reentry, and promoting a healthy workplace environment. Best practice guidelines offer a blueprint for developing a structured substance use disorder policy that includes education, prevention, detection, treatment, and reentry procedures for CRNAs with substance use disorders. Effective substance use disorder policies and procedures that are clearly defined provide illicit responses to situations that place the safety of patients, colleagues, and the individual at risk, as well as the anesthesia group or healthcare facility. I hope you enjoyed my presentation. Thank you.
Hi, let's talk about mentoring and nurse anesthesia. Do mentor nurse anesthetists versus non-mentor nurse anesthetists differ by level of job satisfaction? Hi, I'm Patricia Tharp. I'm a DNP student at Arkansas State University. My mentor is Dr. Christopher Jordan. So let's look at mentoring and nurse anesthesia. Currently, there's an inactive supply of certified registered nurse anesthetists, especially due to the coronavirus. Mentoring is needed in anesthesia in order to increase job satisfaction, increase retention, and prevent turnover. And looking at the review literature, there wasn't a whole lot of literature out there in regard to mentoring nurse anesthesia. Most of it tend to focus on the new RN or F&P. Mentoring itself can change the practice of anesthesia. Now, my question again is, how do mentor nurse anesthetists versus non-mentor nurse anesthetists differ by level of job satisfaction? Let's answer that. My purpose was to measure mentoring among CRNA as it relates to job satisfaction. So I used convenience sample CRNAs working full-time at Gastro 1 Surgery Center in Germantown, Tennessee. My objective was to measure the importance of mentoring as it relates to job satisfaction among CRNAs. So I use Patricia Bennett's theoretical framework. It goes all the way from the novice to the expert. This just goes over her method here. Now, my project design was to implement a mentoring and job satisfaction survey, assess the need for a formal mentor program to improve job satisfaction, retention, and reduce turnover among CRNA and Gastro 1. My methodology: each CRNA received an informed consent, demographic survey, mentor survey, and course in minds of nurse practitioner job satisfaction scale. Data collected was used to assess the importance of mentoring on job satisfaction. This just goes over the tools that I use to measure mentoring and anesthesia. This is my demographic questionnaire, my mentoring questionnaire, and of course my minds of nurse practitioner job satisfaction scale. And it goes on. Now, my methodology: day 1, they were issued informed consent, the mentoring and job satisfaction survey. Days 2 to 5, they were allowed to ask questions. And days 5, I collected my information in a sealed envelope. Now, sampling setting: I used convenience sample of 12 CRNAs employed 40 hours or more at Gastro 1. CRNAs were greater than 25 years of age. Participation was voluntary. Okay, data analysis: I used Excel, SPSS. My 44-item minds of nurse practitioner scale was able to score a 0.96% Cronbach's alpha, which was precisely the same thing as the original scale. Yay! Okay, now this just breaks down my sample. Now, and looking at the data analysis, 100% of the respondents who had a mentor from high positively influenced that mentoring positively influenced their job satisfaction. So we definitely need some more mentoring. This just goes over my analysis. Now, looking at the DNP Essential Doctoral Nursing Practice, I like to think about DNP Essential 1 as the novice going all the way to the expert. That's DNP Essential 8. That's when the novice becomes the expert. Patricia Bennett certainly would agree. Okay, financial budget. Gastro 1 is currently implementing a new 5-day mentoring program for all new high CRNAs. Yay! Now, looking at the costs, mentor nurse anesthetists are more cost effective than non-mentor nurse anesthetists because you don't have to pay for all that turnover. Now, protection of human subjects, each CRNA filled out informed consent. No names or identifying information was given. It was kept on a lock and key. After 30 days, it was shredded. Okay, evaluation plan. All subjects reported that mentoring positively influenced their job satisfaction. So we need some momentum. This finding was shared with the chief CRNA. Gastra 1 is currently implementing a 1-week mentoring program. Yay! Okay. Now, recommendation, I need a larger sample of CRNA, user-friendly, well-based tool to measure mentoring and nurse anesthesia. Lesson learned, explain the purpose of the study to each participant. Recruit as many subjects as possible, and definitely get a mentor. Now, in conclusion, to answer the PICO question, how do mentored nurse anesthetists versus non-mentored nurse anesthetists differ by level of job satisfaction? The answer is mentored nurse anesthetists reported greater levels of job satisfaction than non-mentored nurse anesthetists. So it answered my question. Thank you so much.
My name is Randy Wright, and I am a DNP student at Arkansas State University, and the title of my project is HPV Immunization Intervention Application in Rural Arkansas. The project question is, in males and females age 11 to 18 currently residing in rural Arkansas, does implementation of an evidence-based HPV vaccination clinical intervention program compared with current clinical practice affect HPV vaccine uptake after 1 month of implementation? The aims of my study were to increase HPV vaccination rates, including initiation and completion of series in adolescents 11 to 18 years old in rural Arkansas, and to also increase healthcare provider and clinical staffs practicing in rural Arkansas's knowledge and communication with patients/parents regarding HPV vaccination. I did use descriptive analysis for this study, specifically chi-square test method. Data was exported from Excel to SPSS software, and then everything was numerically coded. These are the formulations that I used for the results of my study. This study was completed in a private rural health clinic in Northeast Arkansas, which provides care for over 7,000 patients, including 600 patients 11 to 18 years old that are eligible for vaccination. The staff includes 2 full-time nurse practitioners and 11 support staff, and then immunizations come from the VFC, SHIP programs, and then private immunizations. The population for the chart review included males and females 11 to 18 years old that had presented to the clinic during the allotted time frame, which was either 4 weeks prior to intervention or 4 weeks after intervention. 100 charts were pulled for both pre- and post-intervention. The survey population included all of the healthcare providers and support staff that worked within the rural health clinic. The instruments used included a survey, which was a validated tool developed by the Gillings School of Global Public Health at the University of North Carolina, Chapel Hill. It was provided pre- and post-intervention to clinic employees, where it was self-administered in a written format. The data collection tool was also used, which looked at gender, age, insurance status, HPV vaccine status, and up to date on other vaccines. The researcher was responsible for creation and application of the interventions and then the training, which included a 30-minute PowerPoint working lunch addressing HPV vaccination and language regarding the vaccine. The intervention bundle included 9 interventions. The interventions included the nurse being responsible for pulling all immunization records for patients prior to the clinic opening daily. All walk-in patients had to have the shot record pulled by the receptionist, and then the vaccine information statements had to be placed on the clipboards with the shot records. Nursing assistants made sure that the immunization record and the VISs were on every clipboard before getting patients back, and the VISs were given to the parents in the room. Nursing assistants had to make sure that the immunization records were placed outside of the room so that the healthcare provider could make sure they were acknowledged. Prior to entering the room, the healthcare provider had to make sure that if there wasn't a shot record on the clipboard, then they had to get it pulled. HPV reminder posters were placed in every patient exam room and in the waiting room. Every patient was scheduled to follow up on appointments for their second or third vaccines, and they were also giving a reminder magnet. An office assistant initiated reminder calls for those that were identified as missed opportunities on WebIZ, and the HPV immunization quality measure was implemented on the EMR system. The results of my study demonstrated that the training and emphasis placed on the HPV vaccine improved clinical staff's feelings about routinely recommending the vaccine. 100% of respondents would recommend the vaccine, and it was important to them post-intervention. Confidence also improved, with 100% feeling confident addressing the HPV vaccine post-intervention. I also found through my survey that the bundled interventions and training were very well accepted, with 100% of the respondents agreeing it fit into their workflow and that they would recommend it to their colleagues. The vaccination rates also were significantly improved, which was an aim of the study. The first injection rate increased 17%, second injection rate 21%, third injection rate 7%, and our series completion increased by 21%. HPV vaccine uptake was directly impacted by this multi-component approach. The results of the study demonstrated significant improvement in HPV vaccination implementation and in series completion, which was the whole aim of the study. The interventions and trainings were well accepted and easily implemented by the staff. And then we're going to see HPV vaccination rates continue to improve as the incidence of the missed opportunities continues to decrease within the clinic. My hope is that future studies are going to focus on utilization of the training and the interventions in other clinic sites.
My name is Ashlyn Lincolnship, and my mentor is Miss Donna Caldwell. For this year's Create at State, I researched a microtranscatheter pacing system. In the United States, more than 2 million people are affected by irregular heart rhythms or arrhythmias, and the most common treatment option for these patients is a pacemaker. Traditional pacemakers work by controlling the electrical activity in the heart, and that corrects arrhythmias. For traditional pacemakers, an incision is made and a pocket is made under the skin on the chest. The pacemaker is then implanted in that pocket and then sutured in. Leads are attached to the pacemaker and they travel through the left subclavian vein into the heart. Well, in 2016, Medtronic, a medical device company, released a new pacing system called the microtranscatheter pacing system, or the micro TPS. This new device is similar to traditional pacemakers in that it does correct arrhythmias. However, that pacemaker is implanted via a catheter through the vein in the patient's thigh, so there's no incision made in the chest and no pocket made under the skin. For this reason, the micro TPS provides a better treatment option to patients in need of single chamber ventricular pacing. I examined infection, complication, and success rates for the traditional pacemaker and the micro TPS, as well as assessed the risk and benefits of both pacemakers. To examine both pacemakers, I evaluated clinical trials and gathered information from Medtronic. Patients who are candidates for the micro pacemaker include patients with atrial fibrillations, or AFib, and symptomatic bradycardia and tachycardia. The micro, however, is not safe for patients with inferior vena cava filters, mechanical tricuspid valves, previously implanted traditional pacemakers, or patients with morbid obesity. The micro is 90% smaller than a traditional pacemaker, and it requires no leads, no incision on the chest, and no pocket under the skin. For this reason, the complication rate is only 2.7%, whereas the complication rate for a traditional pacemaker is 9.4%. The micro has more than a 99% success rate with less than a 1% infection rate. With a traditional pacemaker, there is a surgical scar from the incision that's made on the chest, and the device leaves a visible lump under the skin. Most traditional pacemakers are not magnetic resonance imaging safe, or MRI safe, whereas the micro is MRI safe up to 3 Tesla, which is the strength of the majority of MRI machines in most hospitals. The micro has a minimum battery life of 10 years, and when the battery does expire, a new micro is simply placed next to the previous one. There's no need for surgical removal of the device. A traditional pacemaker has a battery life of anywhere from 5 to 15 years, but when the battery of a traditional pacemaker expires, it requires surgical removal. This is a chest x-ray showing a traditional pacemaker, which is right here, and it shows the leads coming from the pacemaker through the left subclavian vein down into the heart. So here's where the incision is made, and there will be a lump under the skin right there. This is an x-ray showing a micro pacemaker. As you can see, it is much smaller than a traditional pacemaker, and there are no leads anywhere. On the left here is a traditional pacemaker, and on the right here is the micro pacemaker. As you can see, again, it's much smaller, and it's roughly the size of a quarter. Every year, approximately 1 million pacemakers are implanted in the United States. Therefore, it's important to know the different options that are available to patients. And the more informed we are as healthcare professionals, the better we can advocate for better and safer treatment options for our patients.
This is Emily Allen, an MSN Family Nurse Practitioner student. This study is about breast cancer screening rates. I would like to acknowledge Dr. Chris Jordan, my advisor, and Ms. Tanya Mead, my clinical instructor. One out of every 8 American women will develop breast cancer in her life. Breast cancer is the second leading cause of cancer death in women in the United States, with 232,000 women diagnosed with this disease in 2015. The average incidence rate of breast cancer per 100,000 people from 2012 to 2016 was 147.9 for the United States and slightly lower at 142.6 for the state of Arkansas. A screening mammogram is utilized to detect breast cancer at an early stage, which allows for better treatment options and reduced mortality by finding more incidence of the disease while it is still localized and prior to spreading and affecting regional nodes. This supports the practice of providers to encourage all patients to adhere to the recommended mammogram screening guidelines. The United States Preventive Services Task Force recommends that women aged 50 to 74 years old who are not high risk should receive biannual mammogram screenings for breast cancer. The Healthy People 2020 goal for women ages 50 to 74 who have received a mammogram in the last 2 years is 81.1%. Only 72.4% of women in the United States ages 50 to 74 years old are adhering to these screening guidelines. The purpose of this study is to compare the biannual mammogram screening rates of average risk patients aged 50 to 74 in a local clinic in Northeast Arkansas to the national average rate of 72.4% receiving mammogram screenings. IRB approval was obtained through Arkansas State University, and a retrospective chart review was performed for this study. Electronic medical records of 50 patients at a local Northeast Arkansas clinic were reviewed. The dates utilized in the study were January 1st, 2019 through November 18, 2019. A data collection tool was used to gather data. Data collected included age, whether the patient was screened in the office, and if biannual mammogram had been performed. Inclusion criteria were female patients, ages 50 to 74 years old. Exclusion criteria were male patients, any females younger than 50 years old, any female older than 74 years old. If the patient had a current or history of breast cancer, or a first-degree relative with current or history of breast cancer. This study found that the rate of female patients ages 50 to 74 years old at a local clinic in Northeast Arkansas who had received biannual mammogram screenings was 76%. In light of the recommendations by the U.S. Preventive Services Task Force for women ages 50 to 74 years old to receive biannual screening mammograms, this study found that the patients at this clinic had better adherence rates than the United States average, however, still falling below the Healthy People 2020 goal. This leaves room for healthcare providers to address the barriers with some patients or provide a reminder for other patients and further encourage adherence to this guideline. Early detection allows early treatment, which improves the rates of mortality related to breast cancer. This is education that can be provided to our patients. Thank you.
My name is Courtney Andrews Deaton. I'm an MSN FNP student at Arkansas State University. I'm going to discuss my findings on the adherence to hepatitis C screening guidelines within a family practice clinic setting. I would first like to thank my mentor, Dr. Christopher Jordan, and my clinical instructor, Dr. Savannah Coyne, for all their guidance and assistance while conducting my study. Hepatitis C virus, or HCV, is a threat to the health and longevity of American citizens from every state, gender, age, ethnicity, and social background, making early screening and treatment a priority. So much so that in 2012, the United States Preventive Service Task Force made a Grade B recommendation to offer a one-time screening for HCV infection to adults born between 1945 and 1965, also known as the baby boomer generation. The Arkansas Department of Health supports this recommendation to screen baby boomers for HCV in an effort to identify and treat the illness and prevent an increase in chronic liver disease in the future. Hepatitis C virus is the most common chronic bloodborne pathogen in the United States and the leading cause of complications from chronic liver disease. Chronic liver disease is the 10th leading cause of adult death in the United States, and studies indicate that 40% of these cases are HCV related. The CDC reports that while anyone may become infected with HCV, 3 out of 4 Americans infected were born between 1945 and 1965. Persons in this birth cohort have been living with HCV infection for more than 20 years and are reaching the age where detection is critical. The purpose of this study is to evaluate the adherence of a local family practice clinic in Arkansas to the USPST guideline recommendations of providing a one-time screening for HCV to patients born between the years 1945 and 1965, compared to the national average of 13.9%. Prior to conducting the study, IRB approval was obtained through Arkansas State University. The study was conducted by using a retrospective chart review. 50 charts were randomly selected and reviewed through electronic medical records in a rural Northeast Arkansas primary care clinic, and a data collection tool was used to evaluate the data. The collected data included patients seen from November 1st, 2018 to November 1st, 2019, and included age, date of birth, and the CPT code 86803, which is the HCV antibody screening test. My study included patients, male and female, aged 54 to 74 years old, and excluded all patients younger than 54 or older than 74. Here are some other interesting facts about the prevalence of HCV in the baby boomer generation. The CDC reports that this generation is 5 times more likely to be infected with hepatitis C, and that 75% of people diagnosed with hepatitis C were born during this generation. My study found that only 2% of patients, that is 1 out of 50, who met the criteria were actually screened for HCV at this clinic. Compared to the national average of 13.9%, this is an 11.9% variance, and there is much room for improvement. HCV is a slowly progressing disease and may be asymptomatic for decades, leading to a delayed diagnosis and treatment, and may increase severity of the disease at the time of diagnosis. There is no current vaccine for HCV, however, it can be cured. Therefore, it is important to screen for this disease in high-risk populations in order to detect and treat this infection and reduce morbidity and mortality related to its effects. The results of this study show that the USPSTF recommendation of a one-time screening for the baby boomer generation is not being followed. It is important to educate providers about the guideline and encourage screening reminders such as medical alerts in the patient's chart who meet criteria. Again, I'd like to acknowledge Dr. Christopher Jordan, my mentor, and Dr. Savannah Coyne, my clinical instructor. Here are some of my references, and the other references are available upon request. Thank you.
Hello, my name is Charlena Bagwell. I'm a FNP student at A-State. I did an analysis of gestational diabetes screening rates within a local women's clinic setting. This is my poster for my presentation. This is all of the research I've done at the local clinic for gestational diabetes for the screening rates. It talks about the introduction and purpose, the objectives, the methods, the population, the findings, the discussion and action plans, and my references. And I'll talk about all of those individually. Today, gestational diabetes is more common. It has great effect on both mother and the baby throughout the pregnancy. According to an Arkansas PRAMS report in 2012, 10% of women who had a live birth in Arkansas had gestational diabetes. Screening is required within pregnancy between 24 to 28 weeks gestation to diagnose to be able to receive treatment if needed. Gestational diabetes is diabetes that develops during a pregnancy and affects how the cells use sugar. To diagnose the gestational diabetes, an initial glucose challenge test is completed. This is called the 1-hour glucose test. If the blood sugar test is elevated on that glucose challenge, a follow-up glucose tolerance test will be performed. This one's called the 3-hour glucose test. The blood sugars will be checked every hour for 3 hours. If at least 2 of those glucose readings are higher than normal, they're diagnosed with gestational diabetes. With an early diagnosis and treatment of gestational diabetes, complications are reduced that could happen to both the mother and the baby during the pregnancy and delivery. Gestational diabetes can lead to macrosomia, which is a large infant at delivery, preeclampsia, preterm labor, hypoglycemia in the infant, and even delivery of a stillborn. Treatment for gestational diabetes could include diet and lifestyle changes. Oral medication and insulin can help control the gestational diabetes to prevent complications from arising during the pregnancy. The purpose of my study is to raise awareness to the importance of gestational diabetes screening at the appropriate time to receive the needed treatment for a healthy pregnancy and delivery. The objective of my study is to see what percentage of pregnant women are being screened for gestational diabetes within the correct time frame during their pregnancy at a rural Arkansas women's health clinic. This is a picture from the World Diabetes Foundation. It gives details on what gestational diabetes is, how many women it can affect, who is at risk, and what complications that could arise from the diagnosis. For my research, I did a descriptive study that was conducted at a Royal Arkansas clinic through a retrospective chart review of 50 electronic medical records. Simple mathematical calculations were utilized to determine the percentage of patients who were screened at the appropriate time frame in their pregnancy for gestational diabetes. For this study, charts reviewed included pregnant female patients between the ages of 16 to 45 years between the dates of February 2019 and February 2020. This is another picture that goes into detail about the risk factors for gestational diabetes and also the complications that can arise from gestational diabetes. During my research, I found that the women's clinic screened 44 out of 50 of the women, which is 88%, within the appropriate time frame in their pregnancy for gestational diabetes. 4 of these 44 women were screened early due to risk factors in the pregnancy as well within the appropriate time frame. I found that the women's clinic had 6 out of the 50 women that were not screened at the appropriate time frame. This was 12%. 3 of these women did not get screened within the appropriate time frame due to not getting prenatal care. One of these patients had her screening completed at 30 weeks because she did not show up for her scheduled appointment at 28 weeks. And 2 of the women were not screened during the appropriate time frame due to transferring care after the 28-week gestational mark. When considering recommendations to screen all pregnant women between the 24 to 28-week gestational mark for gestational diabetes, the Royal Women's Clinic that I did my research at was found to be efficient in screening women within the recommended time frame. Out of the 50 women in this study, 5 women were actually diagnosed with gestational diabetes and were able to get the recommended treatment throughout the pregnancy and delivery to reduce harm to the mother and baby. My mentor for this presentation was Miss Lauren Hancock, APRN. These are my references that I used for my presentation poster. I hope everyone enjoyed. Thank you.
Hi, my name is Lindsay Beecham. I'm an FNP student, and my presentation is an analysis of depression screening rates within a local urgent care facility that utilizes the PHQ-2 questionnaire. Around 300 million people are affected by depression worldwide, making it a major contributor to the overall burden of disease. 1 in 15 adults will be affected by depression in a given year. The economic burden of this disease is significant for the patient as well as society. Depression is often a comorbidity of other chronic diseases managed in primary care and can impair the patient's compliance and the provider's ability to manage their disease, which makes depression screening and treatment even more essential. Of all the mental health conditions, depression is one of the most treatable, but unfortunately often goes undiagnosed. The USPSTF recommends screening of all adults age 18 or older, regardless of risk factors. And a comparison study showed a national screening rate of 4.2%. The objective of this study was to compare the rates of depression screening in a local urgent care facility in Arkansas to the national average of 4.2%. The study was conducted using a retrospective chart review. Charts were reviewed in an urgent care facility in Hensley, Arkansas. Data was gathered from 50 charts between January 2020 to April 2020. And the depression screen was considered complete if the PHQ-2 was documented on the patient chart in the past 12 months. The inclusion criteria was male or female, age 18 or older, and a new or established patient visit. Exclusion criteria was male or female, less than 18 years of age, or an active diagnosis of depression. 54% of the population was female, and 46% was male. 12% were African Americans, and 88% were Caucasian. The age range for the population was 18 to 81 years old. The findings show of the 50 charts reviewed that 84% of the sample population was screened for depression using the PHQ-2 in the last 12 months. The results show this facility has a much higher depression screening rate than the national average. However, 16% of the sample population still went without proper screening. Limitations of the study were a small sample size with only 50 charts. The facility analyzed was primarily a walk-in clinic for acute care. The data was collected from only one clinic with 2 providers and minimal cultural diversity. Conclusion: the screening rate at this facility is much higher than the comparison study. However, they are not fully compliant with the guidelines set forth by the USPSTF, which recommend all adults be screened for depression. Implications from the study reveal need for further education on importance of depression screening and compliance with the USPSTF guidelines. It includes, they should try to include the PHQ-2 as part of patient intake and establish a way to flag charts in the EMR when there is no screening documented for the past 12 months. And for patients who screen positive, ensure they are diagnosed properly, evidence-based treatment is given, referrals are initiated as needed, and follow-up care is always provided. To conclude, the facility should work towards having an even higher depression screening rate to increase their chances of diagnosing patients and therefore increase the chances for treatment. According to research, 70% of people will improve with proper treatment, making performing screenings that are compliant with guidelines so important. Thank you for your time, and I would also like to thank my faculty mentor, Ms. Hadley, for her guidance during this research project.
Hi, my name is Chelsea Bennett. I'm a family nurse practitioner student at Arkansas State University in Jonesboro, Arkansas. The title of my presentation is A Local Clinic's Rate of Type 2 Diabetics Receiving Annual Comprehensive Foot Exams. I'd like to acknowledge Dr. Christopher Jordan, my advisor, and Dr. Lauren Cood, my clinical instructor. So this is my poster for my presentation. I have the title up at the top, and then I'm going to go through each part of it piece by piece and break it up. According to the American Diabetes Association guidelines, all individuals with diabetes should receive an annual foot exam to identify high-risk foot conditions. Comprehensive foot care that includes screening exams and risk assessments for patients with diabetes reduces the rate of ulceration nearly 75%, and amputation up to 85%. And then diabetic foot ulcers are the leading cause of non-traumatic lower extremity amputation in the U.S., and costs related to the treatment of diabetic foot ulcers are well over $1 million annually. Despite all these staggering statistics, many primary care providers fail to adhere to these guidelines. Data suggests that the diabetic foot is only adequately evaluated only 12 to 20% of the time. So this is just something that I included in on my poster. It has the criteria that the provider would look at on a diabetic foot, and then the risk classification that that patient would fall under with the criteria, and then just patient education, such as the patient should always wear shoes and shouldn't be barefoot, and then they should also evaluate their own feet daily and call their provider with any abnormalities. There's also a few diagrams on here. The purpose of this study was to compare the percentage of adult patients 45 to 75 years of age with type 2 diabetes mellitus that are receiving their annual comprehensive foot exam and a risk assessments in adherence with the American Diabetes Association guidelines in a rural clinic in Northeast Arkansas compared with the national average. First, an IRB was obtained through Arkansas State University. The study was conducted using a retrospective chart review of 50 charts through electronic medical records in a primary care clinic from November 1st, 2018 to November 1st, 2019. A data collection tool was used to gather and transcribe information. The data that was collected included age, gender, ICD-10 code, and if the patient had or had not received a diabetic foot exam. The patient population included both men and women, ages 45 to 75, with a diagnosis of type 2 diabetes, and then they had to be a patient in the rural clinic in Northeast Arkansas from November 2018 to November 2019. This study found that the percentage of foot exams completed on type 2 diabetics from November 2018 to 2019 in the clinic in Northeast Arkansas was 48% compared to the national average of 71.6%. On my poster, I've included two different graphs that show the findings in two different ways, both a bar graph located above the finding section and a pie chart located underneath the finding section. First, I've included the bar graph. The first line is the 48% that the clinic was at for completed foot exams compared to the national average, which is the next line of 71.6%. First, I've included the bar graph. The first line is the 48% that the clinic was at for completed foot exams compared to the national average, which is the next line of 71.6%. This is the pie chart that I was talking about. The blue is the clinic's average of foot exams completed, and then the red is the national average. You can see that the average is significantly more than the clinic's. This study found that the foot exams were performed significantly less often for diabetics given at the local clinic in Northeast Arkansas when compared to the national average. An action plan to fix this could be to encourage the providers to perform the foot exams by showing them this study, or to just something simple like having the person, nurse, or whoever is taking the patient back, have them take off their shoes and their socks if they're diabetic. That way the provider knows that that needs to be done. So this is my reference page for my presentation. Thank you for listening.
This presentation is titled An Analysis of Adolescent Depression Screening Rates in Primary Care. I would like to thank my mentors, Dr. Mark Foster, Dr. Christopher Jordan, Ms. Atisha Hadley, and Ms. Lauren Hancock for their help with this project. Adolescent depression is on the rise in the United States. The purpose of the study is to raise awareness about the rates of adolescent depression and provide information about what can be done to help. The CDC reports that 1 out of 20 adolescents from the ages of 12 to 18 have been diagnosed with depression. Depression that begins during the adolescent time period often leads to drug use and addiction, low self-esteem, poor school performance, and suicide. When an adolescent experiences depression, it often lingers into adulthood and gets more difficult to treat. This leads to lifelong problems. The United States Preventive Task Force makes recommendations for services to improve standards of care in the healthcare environment. The United States Preventative Task Force recommends screening 100% of the adolescents between the ages of 12 to 18 for depression annually. The task force also recommends that an adolescent that is found to be depressed needs to get appropriate treatment, referrals, and a follow-up should be made. Treatment may include medication and/or therapy. With medication or therapy, appropriate follow-up should be implemented in order to provide seamless care for the patient. This particular study was completed by conducting a retrospective chart review in a family practice clinic in rural Arkansas. There were 40 charts that were reviewed of adolescents from ages 12 to 18. The data was collected from August 2019 to March of 2020. In this particular clinic, they asked every patient between the ages of 12 and 18 that came in for their annual visit if they had symptoms of depression. So on the surface, it looks like they're meeting the 100% recommendation, but there are some issues to address. The clinic did not fully utilize the PHQ-9 form, which is a depression screening tool. It has a list of questions and can be filled out discreetly, which may sometimes be easier for an adolescent than just answering straightforward questions. Also, the clinic lacked a follow-up plan. If the patient with depression was placed on an antidepressant medication, the provider did follow up. But if the adolescent patient was just referred to a therapist, there was rarely follow-up to ensure treatment. Ensuring follow-up creates seamless care for the patient, and it encourages adequate treatment. The PHQ-9 form and following up by managing depression are both billable services that would compensate the clinic. The goals are for this clinic to utilize the PHQ-9 form for every adolescent between the ages of 12 and 18, to educate the staff on exploring and addressing depressive symptoms in patients, and creating a plan for better follow-up after a mental health referral. Overall, implementing these steps would help identify and treat adolescent depression better in the primary care setting.
Good afternoon, everyone. My name is Madison Ferguson. I am a nurse practitioner student at Arkansas State University. The research I have conducted is an analysis of childhood obesity screening rates among a suburban pediatric population. The mentors I've had throughout this process are Dr. Mark Foster, Dr. Christopher Jordan, Ms. Otisha Hadley, and Ms. Lauren Hancock. I hope you all enjoy. So this is an at-large view of the poster I created for my research. Throughout this presentation, I will be discussing each subtopic individually. With having a nursing background in pediatrics, working in Memphis, Tennessee, this is a topic I've become very passionate about, so I truly do hope that you enjoy. Okay, so let me give you a little background regarding childhood obesity and why it is of such importance. The prevalence of childhood obesity in the United States is about 18.5%, affecting nearly 13.7 million children and adolescents. We can narrow that number a little further, with research showing that children aged 10 to 17%, 31% of them are either overweight or obese. The percentage in Tennessee is actually higher than that, with 36% of their youth falling into that category. Now, why is this important? Well, obesity in childhood years is more likely to lead to multiple complications and disease processes as they reach adulthood. This includes adult obesity, cardiovascular disease, diabetes, musculoskeletal disorders, and even puts them at a greater risk of developing some forms of cancer. In an attempt to combat these statistics, the U.S. Preventative Service Task Force released a Grade B recommendation that all clinicians screen for obesity in children and adolescents over the age of 6 by the means of BMI, and then offer or refer them for comprehensive, intensive, and behavioral interventions to promote improvements in weight status. Research was conducted and showed that on the national level, about 88% were doing the annual screening. However, only 64% were offering some form of intervention, with only 7% offering intensive interventions that were recommended. The objective of this study was to compare the screening practices of the providers in a pediatric clinic in Southwest Tennessee with the national average discussed above. I then took one step further, analyzing and comparing interventions on those who received a positive screening for obesity to the national level. I narrowed my research parameters to only be conducted on children over the age of 6 who would present to the clinic for their annual well-child visit. From there, 50 random electronic medical records were selected, and a retrospective chart review was performed. When compared to the national average of 88%, the clinic surpassed that standard, screening 98% of their patients for obesity by the means of BMI. Of these patients that were screened, 52% were either overweight or obese. 30% screened positive for obesity, with a BMI over the 95th percentile. Of those who were considered obese, the clinicians did provide education regarding a healthy diet, limited screen time, and proper exercise. ICD-10 code of Z71.3, which is Dietary Surveillance and Counseling, was built on 100% of these visits. ICD-10 code Z71.82, which is Exercise Counseling, was only built on approximately 33%. Now, of those who were considered to be obese, only 6.7 of these children and or their parents were provided the recommended interventions or referrals recommended by the task force. This was slightly less than the national average of 7%. After reviewing these charts, I realized that there are many billable codes that can maximize reimbursement for these providers that were not being utilized. Education can and should be done on all of our patients, regardless of their BMI status. Therefore, services should be billed for. I have listed the codes below for your observation. Now, what are these intensive interventions that I've been speaking of? The task force conducted research that proved that greater than or equal to 26 contact hours, targeting both the parent and the child, either separately or together, and providing education regarding healthy eating, safe exercising, reading food labels, stimulus control, goal setting, rewarding, and supervised physical activity, resulted in weight loss. If done by the practitioner, it is billable, and I have provided the billable codes below. Now, keep in mind this is often a multidisciplinary team approach, including pediatricians, physical therapists, psychologists, and dietitians. We should utilize these resources and our colleagues for the health of our patients. My goal in conducting this research was to address the need of yearly obesity screening and the importance of initiating an appropriate intervention plan to combat the childhood obesity epidemic. I want to thank you all for taking the time to listen to my presentation. May you have a blessed day.
Hello, all you Red Wolf fans. My name is Frances Butler. I'm a senior family nurse practitioner student with the College of Nursing and Health Professions here at ASU. My project title is Our Rural Primary Care Clinics Adhering to the National Guideline of Non-Pharmacological Treatments as First-Line Care of Low Back Pain. My mentor for this project was Dr. Christopher Jordan. I'd like to start out my presentation by giving you all some facts about back pain. It's the primary contributor for missed work days and the leading cause of disability in Arkansas, as well as worldwide. It is the most frequent reason for healthcare visits and the most common reason for opioid pain treatment. Now, this trend to treat pain with opioids began back in the late 1990s when pharmaceutical companies encouraged providers and clinicians to prescribe opioids for pain, claiming that they were not addictive. However, we now know that they are extremely addictive. As a matter of fact, in 2017, 1.7 million Americans claimed to have an addiction to opioid painkillers, and these are prescribed painkillers. And unfortunately, also in 2017, 47,000 Americans died of opioid overdoses. 188 of those were Arkansans. So to fight this epidemic, the American College of Physicians released a new clinical practice guideline. This guideline called for first-line treatment, which means the first time they come into the office with low back pain, they should be treated with non-pharmacological methods instead of opioids. So the purpose of my study is to compare an Arkansas clinic's first-line treatment of low back pain with the first-line treatment in the Northwest U.S., consisting of physical therapy at 8.7% and opioid prescriptions at 25.3%. This is a slide showing the comparison study with physical therapy being prescribed at 8.7% and opioids at 25.3%. The objective of my study was to determine whether first-line treatment practices of low back pain with opioids at an Arkansas rural clinic, if they were better than those of the Northwest U.S. Now, what I wanted to do with this information was increase the provider's knowledge of how they practice, of their practice patterns in the clinic, so that if they needed to, they could set future goals for improvement. That way, it could help with the opioid crisis in America. Prior to data collection, I did receive permission from ASU's IRB and from the site where the data was collected. The method I used for my project was a retrospective chart review of 50 electronic medical records that was conducted at a rural Arkansas clinic. Now, the charts that were reviewed included both male and female patients between the ages of 18 and 64 with an ICD-10 code of M54.5. These patients visited the clinic between November the 1st of 2018 through November 18th of 2019. I did exclude some patients from this study, and those were patients under the age of 18 and over the age of 64, and those without an ICD-10 code of M54.5. To obtain my findings, I used simple mathematical calculations. I used those calculations to determine the percentage of patients receiving physical therapy versus opioids as first-line treatment of low back pain. Out of the 50 charts that were pulled for my study, only 32 of the patients met the criteria for the project. 0% of those patients received physical therapy, and only one patient, which came to 3.1%, was prescribed an opioid. All other patients, which was 96.9% of patients, received other medications, including muscle relaxers, steroids, or NSAIDs. The rural Arkansas clinic did not meet the new guidelines set forth by the American College of Physicians to treat low back pain. However, the clinic's opioid prescription rate was found to be lower at 3.1% than those prescribing rates in the northwestern United States, coming in at 25.3%. Ultimately, non-pharmacological treatment has been proven to be better treatment for low back pain. The clinic was at 0%, leaving room for 100% improvement. Although the guideline is not being met, they are performing significantly better than the northwest part of the U.S. in opioid prescribing habits, which reduces their patient chances of opioid overdose.
Hello, my name is Tony Cecil, and I'm a second-year student in the Master of Athletic Training program. The research I'm presenting is over the effect of whole-body reactive agility training on volleyball player performance. My faculty mentor for my research is Dr. Scott L. Bruce, and my co-authors are Dr. Robert Bradley and Dr. Carlita M. Moore. Whole-body reactive agility is an important quality for volleyball athletes. Their reaction time, which is the period between a stimulus and a response to that signal, is one of the most important aspects in the sport. Elite athletes, such as intercollegiate volleyball players, can react accurately and quickly to visual cues, such as having the ball spike towards them or a tip being pushed over the net. For my research, I use Tracer technology, which uses computer-based simulation to create an accurate analog of the functional challenges of sports, work, leisure, and daily living activities. It uses full-body sensing and projects an avatar of the participant to measure analytics to provide information from the athlete. Baseline and post-test involved 4 activities: lateral agility, which is when athletes move side to side based on target appearance, lateral flanker, which is when athletes respond to a central arrow direction to hit a virtual target, moving right to left. Diagonal agility is when athletes move back and to the right or left based on target appearance, and diagonal flanker, which is when athletes respond to a central arrow direction to hit a virtual target, moving back right or left. Single task and dual task training was involved throughout the testing. Single task training is when targets show up until they are hit without any secondary task requirement. Dual task training, which is also considered the flanker test, is when a central arrow direction dictates the motor responses. The purpose of this study was to train NCAA Division I volleyball players with Tracer to determine if a positive change in volleyball game statistics was possible. For my study, 15 intercollegiate volleyball players volunteered to take part. They were baseline tested on the Tracer prior to the 2019 season to assess their whole body reactive agility. After baseline testing, the volleyball players were trained on the Tracer twice per week for 3 weeks with a variety of 3 exercises. These training sessions lasted about 5 to 7 minutes per session, depending on which exercises they were doing for that training session. Once the 3 weeks of training was complete, the volleyball players were post-tested by repeating the baseline tests. Independent t-tests were ran and used to compare the Tracer variables and game statistics. Paired t-tests were also used to assess the differences between baseline tests and post-tests. With the results of the study, there were no statistically significant changes to volleyball game statistics found. There was poor statistical significance and correlations between Tracer data and volleyball game statistics, which is being attributed to the fact that the Tracer training only lasted 3 weeks. The table that is presented is of the paired t-test results, with only 8 of the parameters being statistically significant. However, there were several positive outcomes and improvement with the Tracer training. Lateral agility saw 66.7% of the athletes improve from baseline to post-test. Diagonal flanker saw 64.3% of the players improved over the course of the training. 35.7% of the players improved on the lateral flanker. Diagonal agility saw 46.2% of players improve from baseline to post-test. 83.3% of the players' reaction time improved, and time differences between right and left directions known as asymmetries improved by 81.3%. Training on the Tracer for only 3 weeks produced several positive changes and clinically significant results. Future research ideas could include increasing the length of the training time, increasing how long each session lasted, and figuring out how long throughout the season the training should last.
Hello, my name is Stacia Coleman, and I am a second-year athletic training graduate student. My mentors for my research are my professors, Dr. Scott Bruce, Dr. Carlita Moore, and Master in Athletic Training Program Director, Dr. Robert Bradley. My research is on the predictive value of injury risk screening for intercollegiate basketball players. The purpose of this study was to determine what factors of the baseline testing predicted musculoskeletal injuries in NCAA Division I basketball players. While this is not the original purpose of the study, my purpose had to be revised due to the coronavirus pandemic. This study uses visual motor reaction time, or VMRT, which is assessed and measured using the newest light board technology available, the reflection edge. Participants also answered an 11-question version of the Sport Fitness Index, or SFI for short, which quantifies persisting effects of previous musculoskeletal injuries on a 0 to 100-point scale, of which question 11 asks about any concussion-related symptoms the participant is currently suffering from. The outcome variable for the study was musculoskeletal injuries, which we defined as any orthopedic condition that was treated and documented by athletic trainers during the course of the regular season. We then used these tests along with games started and games played to create a baseline test for our participants, where we aim to determine which of these factors can predict what athletes may be susceptible to musculoskeletal injuries. While there is research that uses prediction models to determine which athlete is likely to suffer a musculoskeletal injury, what makes this research so unique is that, to our knowledge, there is no published study focusing specifically on basketball players. The participants of this study consist of 30 NCAA Division I basketball players, 16 women's basketball players, and 14 men's basketball players. For this study, VMRT is measured using the reflection edge, a 2600 LED light board displayed in Figure 3, on which participants perform 3 tests, referred to as Big G, where a participant touches illuminated targets as quickly as possible, similar to the game of whack-a-mole; Go No Go, where the participant was instructed to ignore red targets, touching only green targets; and Right and Left Execution, or rail, where the participants were instructed to hit yellow targets with their left hand and purple targets with their right hand. The light targets are illuminated one at a time and deactivated by touch. After VMRT was collected and SFI was quantified from the preseason, we analyzed game exposure and injury data upon the completion of the regular season. This allowed us to derive a multivariable prediction model to determine the predictive power of the variables. This includes receiver operating characteristic, or ROC analysis, to determine cut points for binary categorization. The binary predictor variables were crossed with injury occurrence, represented by a univariable odds ratio with 95% confidence intervals, at which point we use logical regression analysis to identify the strongest set of binary variables, which provide the final model for the ROC analysis, producing a number of positive risk factors. From the study, our initial model was created using data from all the players, which produced the 4-factor model. The 4 factors are the SF5 question 11, games played, games started, and go no go average motor time, which was applied to each team individually. For the women's team, all 4 factors were in the model, with any combination of 3 or more factors being the optimal number of predictors, indicating that a player with 3 or more factors had 20 times greater odds of suffering a musculoskeletal injury compared to those with less than 3 factors. For the men's team, games played and games started yielded the same result, so a 3-factor model was analyzed and 2 or more factors was optimal. The odds ratio of 0.75 indicated the odds of a player with 2 or more factors indicated that they were less likely to get injured compared to those with less than 2 factors. While the specific factors in the cut point may differ from team to team, I would recommend that athletic trainers repeat the study using the SFI-11 and reflection edge to identify which athletes are considered high risk of injury and implement remedial programming in an attempt to prevent future musculoskeletal injuries.
Hi, my name is Ashley Davis. I'm an FNP student at ASU. I did a research project over mammogram screenings, and it's titled A Comparison Analysis of Women Ages 50 to 74 that have had mammograms within the primary care setting. This is the poster I made for Create a State, and in the following slides, I will talk about each section a little more in depth, describing the parts of the research project. The introduction and purpose just starts talking about breast cancer in general. Breast cancer is the most common cancer in women and the second most frequently diagnosed malignant tumor worldwide, with an incidence of about 12%. Each year in the U.S., about 245,000 cases of breast cancer are diagnosed in women and about 2,200 in men. Of those, 41,000 women and 460 men in the U.S. die each year from breast cancer. Having mammograms is associated with a 19% reduction in breast cancer mortality rates, which is why it's a good idea to get them done. In 2009, the United States Preventive Services Task Force made new recommendations for women ages 50 to 74, recommending routine mammography every 2 years. According to the National Cancer Institute, the latest statistics show that 71.6% of women aged 50 to 74 years had a mammogram within the past 2 years. The primary objective of this study is to compare the percentage of women who are having mammograms routinely ordered at a family practice clinic in Paragould, Arkansas, to the national average of the mammograms ordered. By evaluating the results of this study, it is determined whether the clinicians in the family practice clinic are ordering mammograms more than or less than the national average. The results will shed light on the provider's performance, and they can use the data to make improvements in their use of screening tools. This was a descriptive study done at a family practice clinic in Arkansas through a retrospective chart review. We used 50 electronic health records of women ages 50 to 74 that were seen in that clinic. Simple mathematical calculations were used to figure the percentage of those women who had a mammogram ordered. The charts I reviewed in the clinic included women ages 50 to 74 who had been seen at the clinic between the dates of May 1st, 2019 and November 30th, 2019. Patients that were excluded from the study were men, women who were younger than 50 or older than 74, and women who had had a previous bilateral mastectomy. As you can see in the graph, the study found the rate of mammograms ordered in this family practice clinic for qualifying women was 72%, leaving 28% of women who qualified that did not have them ordered. When reviewing recommendations by the task force to give women 50 to 74 years of age biannual breast cancer screenings, the family practice clinic in Arkansas's rate of mammograms ordered was almost the exact same as the national average. They had 72%, the national average is 71.6%. Based on the evidence of proven research, mammograms do decrease the morbidity and mortality of breast cancer. Knowing this and how easily achievable this is by ordering at a visit such as a yearly wellness, it is still not being done on every woman in this age group. More than 25% of qualifying women do not receive breast cancer screenings as recommended by the task force in the family practice clinic or in the nation. There's definitely room for improvement in ordering screenings to decrease the risk of breast cancer mortality. The last two slides is an acknowledgement to the faculty of the Family Nurse Practitioner program at ASU, as well as the references used for this project.
Evaluation of metformin prescribing rates compared to national rates by Kelly Dowden. Faculty mentor, Dr. Nancy Baltz. This is my poster that I created for Create a State. The following PowerPoints will describe the Quality Improvement Project. There are 34.2 million Americans living with type 2 diabetes. Uncontrolled hyperglycemic levels can cause irreversible heart, kidney, eye, and nerve damage. Primary care providers deliver care to 90% of the type 2 diabetes patients. Metformin is the first-line drug used to treat type 2 diabetes patients because it is cost-effective and decreases hepatic glucose production without causing hypoglycemia. Despite the benefits of metformin, only 65% of the type 2 diabetes patients are prescribed the medication. The purpose of this study is to determine the percentage of type 2 diabetes patients aged 18 to 70 in an urban primary care clinic were prescribed metformin, according to the American Diabetes Association and the American College of Clinical Endocrinologists. For this study, institutional review board approval was granted by Arkansas State University. A retrospective chart review of 50 patients with the diagnosis of type 2 diabetes seen in an urban clinic in Memphis, Tennessee. A data collection tool was used to gather and transcribe the data. And descriptive statistics were used to analyze and compare data using Microsoft Excel. Inclusion criteria included a diagnosis of type 2 diabetes. Patient could be male or female, age 18 to 70, and the patient must have been seen in the clinic between November the 1st, 2018 and November the 1st, 2019. Of the 50 charts audited, 43, or 86%, of the patients were treated according to the American Diabetes Association and the American College of Clinical Endocrinologists guidelines. Of the 43 patients treated with metformin, 32, or 73%, had controlled blood glucose levels. The patient's age ranged from 32 to 69, with the average age of 55. There was a 21% variance between primary care and the national average. Although the study shows an 86% medication adherence rate to the American Diabetes Association and the American College of Clinical Endocrinologists guidelines among type 2 diabetes patients, there was still a significant number of patients with uncontrolled blood glucose levels. The advanced practice registered nurse has an opportunity with each patient encounter to discuss the management of blood glucose levels. Discussion should focus on medication adherence, in addition to encouraging physical activity, which can aid in decreasing blood glucose, weight loss, lowering blood pressure, decreasing cardiovascular risks, and decreasing depression. Limitations to this quality improvement study include a small sample size of 50 charts, and only one clinic was used for data collection. Additional references available upon request.